Hello, everyone. Welcome to the very first joint symposium of Kyoto University's 3 North American on-site laboratories. And we have a wonderful title, Transformative Innovations in Medical and Life Sciences. And my name is Kaoru Kitajima. I will be the overall moderator, but we have people who are chairing several, actually 3 sessions plus opening and closing keynote. And so without further ado, we have a packed program. So we appreciate everyone to be helping us have a timely and exciting symposium. Before we begin, I'd like everyone to turn off your cell phone, or put it in silence mode if you haven't done so yet. And we have a lot of lectures and short Q&A sessions, etc. We really need to have the whole thing running and in a timely manner. And I would like now to call Professor and President of Kyoto University, Dr. Nagahiro Minato, and he will give a short welcoming speech. Dr. Minato, please.
Good afternoon, distinguished guests, colleagues, and friends. Thank you for joining us today, whether you are participating in person or online. As President of Kyoto University, I'm pleased to welcome all of you to the first joint symposium of Kyoto University's 3 North American on-site labs. I would like to convey special gratitude to Dr. Kosler, Chancellor of UCSD, with whom I talked through time this morning, and Dr. Peek Acer, UCSD's Vice Chancellor for Research, Dr. Kersaz, Vice Chancellor for Health Sciences, all of our colleagues at the UCSD, and all of the other researchers and institutions for their great support toward holding this symposium. Kyoto University celebrated its 125th anniversary last year. Since its founding in 1897, the university has been committed to the principle of academic freedom and pursuit of research and education to promote harmonious coexistence among members of global community. As part of the mission, the university seeks solutions to the complex challenges of the modern world, making efforts to share its research findings with society and responding to the ever-challenging demands of the time. Since the launch of the on-site laboratory initiative in 2018, Kyoto University has established 11 laboratories in collaboration with overseas partners to promote advanced research, international education, and academia-industry collaboration. 3 of those 11 on-site laboratories were established in collaboration with North American institutions: the University of California, San Diego, the Gladstone Institute, and the University of California, Los Angeles. And today, for the first time since their establishment, the 3 North American-based laboratories are holding a joint symposium. In 2018, Graduate School of Medicine established on-site laboratory at UCSD's Center for Novel Therapeutics. This laboratory aims to promote joint research and share state-of-art innovation and new technologies in the field of science, life science, collaborating with startup biotechs and medical science companies. As an extension of its mission, showcase, our Kyoto University Life Science Showcase at San Diego has been hosted by every year, I guess, since 2020. The latest edition of the event took place actually yesterday in this place. The IPSL Research Center at Gladstone Institute was established in San Francisco in 2019 as a reflection of Kyoto University's long-standing collaboration with Gladstone. The center aims to achieve groundbreaking discoveries through biomedical research on iPS cells to develop its new methods to treat human diseases. We have Shinya Yamanaka. He is presenting part of this story today. The Quantum Nanomedicine Research Center was also established in 2019 in collaboration with UCLA. And located in Kyoto, the center aims to merge physics and medicine to pioneer a new academic field of quantum nanomedicine, taking advantage of quantum beam radiation and nanomaterials technologies. Building upon these continued collaborations and partnerships, this symposium will address selected themes that highlight socially significant research and innovations in medical and life sciences. I hope that today's event will provide an opportunity to share cutting-edge research findings surrounding the keywords of stem cells, reprogramming, quantum nanoscience, and cancer immunotherapy. I sincerely hope that today's event will enrich academic networks between Japan, the US, and beyond, enabling us to work together and contribute to a better future for humanity. In closing, I would like to express my sincere appreciation once again to the speakers, chairs, and all of the participating researchers, institutions, and administrative staff for their efforts toward holding this scientifically remarkable event. Thank you very much, and enjoy your symposium.
Thank you, Dr. Minato. I now would like to invite Dr. Corinne Picasa of UCSD for a brief welcome remarks. Thank you.
Thank you. On behalf of Chancellor Khosla, I'm Corinne Picasa, the Vice Chancellor for Research, and I want to say a very big welcome to colleagues coming from far and near, for colleagues here with us today, as well as colleagues remotely joining us. It's a very exciting time to build on this relationship, which is long-standing. I think there's so much more exciting collaboration to come. And overall, it's an exciting day for science. There has never been a time when the work we do can be so transformative for lives of the people that we serve in doing our research. And I think that it's easy to come to rooms like this full of scientists and take for granted that our research sometimes feels slow, it's very cerebral, but the advances that you're making will truly improve lives of patients and their families who can be devastated by the diseases that your work is helping to cure. So pat yourselves on the back for just a moment. And then also take great pride in the ability to come together. We haven't been able to be in person for quite a while, but these exchanges where we have the presentations of laudable minds, the ability to talk over ideas over coffee, are really the essence of creativity that you all want in our scientific discovery process. So please take the time to enjoy each other's company. It's also an exciting time in science because we have never seen the opportunity to collaborate in the area of innovation, commercialization, and technology development to solve the world's big health problems in a way that we do today. And so President Minako had examples from the partnerships they have internationally that include not only the breadth of health sciences, but engineering, physics, quantum computing, materials science. So bringing all of those perspectives together is something that really happens in these magical collaborative partnerships. As one example, with our focus with Kyoto University on regenerative medicine and cancer immunotherapies, our radiology department will host a group of colleagues from Kyoto University to explore our UCSD Molecular Imaging Center and the Center for the Future of Robotics to bring together interdisciplinary approaches to understand how imaging can impact and improve our precision in cancer surgery. So we're very excited about that as an example of many potential exchanges. One more reason that this is such an exciting time for science is that we are in a very challenged world. We have challenges in politics, we have challenges in climate change, we have challenges in economics, all outside the realm of health. But there have never been more important relationships than those between academic institutions when we want to successfully tackle some of our greater social challenges. So I don't want to put too much of solving the problems on the world on your shoulders, but I will say that the value to us of our international collaborators is so much more than the science. It's the interpersonal, it's the stability, it's the creativity and the discovery. So we celebrate all of those wonderful things together today. Thank you.
Thank you very much, Vice Chancellor Picasa. Now I would like to introduce the chair of the opening keynote session, Dr. Juan Carlos Belmonte. Dr. Belmonte is the director of San Diego Institute of Science Altos Lab. Dr. Belmonte, please.
Thank you for your introduction. Good morning, everyone. It gives me an enormous pleasure to introduce Dr. Shinya Yamanaka. Shinya has been the key person in my research career, research interest, research question that has modeled most of the work that we have done in my lab. Thus, this pleasure to introduce him today to you. Shinya is the President Emeritus of the Sara Institute in Kyoto, as well as a professor in Kyoto. And as you all know, his conceptual discoveries of how a specific set of transcription factors can de-differentiate cells to a more plastic state has really revolutionized and perhaps pioneered this new brand of medicine, regenerative medicine, which is one of the key topics of this symposium. So again, many thanks, Shinya, for your work, and the podium is up to you. Thank you very much.
Thank you very much, Juan Carlos, for your very kind introduction. Good afternoon, everyone. My name is Shinya Yamanaka. Actually, San Diego is one of my favorite cities in the States and also in the world. I landed like 3 hours ago from Japan, but I already enjoyed running, and I was able to go down to this beautiful beach, so I was happy. But on the way home, I had to climb up this steep hill amid jet lag, so I guess no pain, no gain. So I have a long history with California, actually, not San Diego, but San Francisco. I did my postdoc training in 90s, like almost 30 years ago at Gladstone Institute in San Francisco. And at that time, I made some unexpected result. So this is the summary I did in my postdoc training. Here's myself and my mentor, Tom Minerality. You can see I had a bit more hair at this time. So, I'm sorry. we were working on ApoBIC1. At that time, we thought ApoBIC1 was RNA editing enzyme, but it is more famous as DNA mutating enzyme nowadays. But I made one unexpected discovery that overexpression of ApoBIC1 in transgenic mouse liver caused hepatic tumorigenicity, hepatocellular carcinoma. So I continued working on hepatic tumors, and I identified one novel gene, novel target of ApoBIC1, which I designated NAT1 as a candidate gene responsible for tumorigenicity by ApoBIC1. Then I made knockout mice and also knockout ES cells and found that NAT1 is essential for early mouse development and ES cell differentiation. Because of these series of unexpected results, I got very, very interested in ES cells, pluripotency, and that resulted in the development of iPS cells back in Japan in 2006 in mouse and 2007 in human. So all we need is a combination of 3 or 4 transcription factors to convert skin or other blood cells, somatic cells, back into the embryonic state, and to make induced pluripotent stem cells, iPS cells. So soon after we reported the generation of human iPS cells, Kyoto University decided to start a new research institute for the development of iPS cell research and application. That is CIRA. CIRA has a very clear mission. It is to bring iPS cells to patients. So there are 2 major medical applications of iPS cells shown here. So once we make iPS cells, we can expand iPS cells as much as we want. Then we can convert iPS cells into virtually all types of cells, such as neurons, heart cells, muscle cells, hepatic cells, and so on. So we could transplant those cells back into patients, for example, suffering from some neurological diseases. We should be able to recover the functions, cellular functions in patients. So this approach is called regenerative medicine or cell therapy. Another important application of iPS cells is that to use these various types of human cells in laboratories, in academia, or in pharmaceutical companies, to recapitulate diseases in dishes, and also to search for effective drugs to stop disease progress. So these are the 2 major applications which CIRA has been working on. So let me give you a brief update on each of these 2 major medical applications, and let me start with regenerative medicine. The ideal usage of iPS cells in regenerative medicine is to use patients' own iPS cells, autologous iPS cell transplantation. However, the process to use patients' own iPS cells is a very, very long process. And we found it is too expensive and too much time consuming. At the moment, it costs up to $1 million for one patient, and it could take up to 1 year at the moment. So it is not practical, I have to say, it is not practical to use patients' own iPS cells in cell therapy. at this moment. So as an alternative approach, we have been working on this project, iPSL stock for regenerative medicine. So this is an allograft. We use iPSLs from donors. So we need to match HLA to reduce immunogenicity. So this is our strategy to minimize immune rejection. We decided to use HLA homozygous donors. So in this slide, I show, in the upper panel, upper portion, I show the HLA haplotypes of 10 patients by different colors. We received one color from our father and another color from our mother, and the combination makes it more diverse. None of you in this audience probably has the identical HLA haplotypes. So if we want to prepare iPSL stock for these 10 individuals, we need to establish 10 lines from all HLA haplotypes. However, if we identify one such HLA homozygous donor who happened to receive the same color, in this case red, from his or her mother and father, the situation is very different. 4 out of 10 patients can be treated with this just one HLA homozygous donor, because these 4 individuals, so these 4 individuals shown by the arrows have red and something else. So as long as he or she receives red from HLA homozygous donor, it's okay. So this is our strategy to reduce the cost and time of autologous iPSL generation. Of course, our immune system is much more sophisticated. For example, if this person who has red and green, and if he or she receives only red, this person, the immune system of this person should think, well, red is okay, but green is missing, so something is wrong. So we need to worry about that kind of missing self immune response mainly by NK cells. But nevertheless, by matching HLA, we believe that we can significantly lower immune rejection reaction. So this is how we actually generate iPS cells from HLA homozygous donors. We need to recruit HLA homozygous donors. It's not so common. Only 1 out of 500 or 1 out of 1,000 people. But we are getting a huge support from Japan Red Cross. They have multiple programs such as platelet transfusion, bone marrow transplantation, and also cord blood banks, with which they have established a huge database of the haplotypes in Japanese population. So by obtaining a huge support from Japan Red Cross Society, we have been able to identify HLA homozygous donors very effectively. And once we receive consensus from these donors, we make clinical grade, I would say GMP grade iPS cells in cell processing facility in our institute. This project has been taken over by Saira Foundation, which I will describe later in my talk. So we started distribution of our first HLA homozygous line in 2015, almost 8 years ago. This project has been led by this person, Naoko Takasu. So we have distributed our lines to many people in academia and also in industry, mainly in Japan, but in some foreign institutes as well. And they are now being used in clinical trials. Actually, many, more than 10 clinical trials are ongoing in Japan using our iPSL stock. The first one is Parkinson's disease application, which our colleague Jun Takahashi will talk about later today. But in addition to Parkinson's disease, we have many eye applications, both retinal and corneal applications, some tumor applications. Scientists are making immune cells attacking cancer cells from iPS cells. We have cardiac applications, blood applications, like spinal cord injuries, and cartilage defect. These are all ongoing. And a few others, like type 1 diabetes and solid cancers applications, these are being planned. So I would say so far so good. We need to carefully observe the outcomes of these ongoing clinical trials. But one thing I want to say is that in these ongoing clinical trials, in all cases, people are using two-dimensional cells, like brain cells, heart cells, in most cases. So this is the current cell therapy. But many scientists are already moving forward. Many scientists are generating 3D structures, such as tissue, like organoids, mini organs, and organs itself by utilizing like pigs. And also one extremely interesting and important new technology is rejuvenation by partial reprogramming. So we have shown that we can make iPS cells from like young somatic cells, but also we can make iPS cells from centenarians, 100-year-old. And once they become iPS cells, they are very young. Judging from DNA methylation status, their age is almost 0. So we can convert young, like 20-year-old fibroblast or 100-year-old fibroblast back to 1-year-old fibroblast, or I mean iPS cells. So the question is, can we do this? Instead of making iPS cells, can we rejuvenate old somatic cells by utilizing like the same factor back to young somatic cells? This may sound like science fiction, but it's already happening. And Juan Carlos is going to talk about this kind of approach very soon. So let me move on to the second equally important application of iPS cells, which is drug development. Because of the interest of time, I don't give you any specific examples, but for this second important applications, multiple clinical trials are already ongoing in Japan. And I'm sure many others are also ongoing in US and in other countries as well. But I only show clinical trials in Japan. We have one clinical trial for Alzheimer's disease, one for Pendred syndrome, one for fibrodysplasia ossificans progressiva, FOP, and we have two for amyotrophic lateral sclerosis, ALS. One by Haruhisa Inoue of Kyoto University, and the other is by Hideyuki Okano of Keio University. So in all applications, people have conducted phenotypic screening using iPSL-based model. And then they studied existing drugs. And they found some existing drugs are effective in Petri dish. Then, because they are using existing drug, it's kind of a drug repurposing. It took very short to move from iPSLs to clinical trial. In the case of ALS by our colleague Haruhisa Inoue, he published iPSL result in 2017, and he started a clinical trial in 2019. So it took only 2 years. And then he had a very promising result from phase 1 clinical trial, and he published the result of phase 1 clinical trial already, and he's now conducting phase 2 clinical trial. So once again, so far so good. But this is probably the best we can do. We means scientists in academia. We can bring iPSL or any new technologies to a very small number of patients, like 10 patients, 5 patients, through this kind of small-scale clinical trial. But that's not our goal. We want to bring our technologies to thousands of patients suffering from intractable diseases. So we, academia alone, cannot complete that task. We need a huge support from industry, especially big pharmaceutical companies. But as you know, there is a huge gap between academia and industry, and this gap is often called valley of death. We really have to overcome this valley of death. Otherwise, drug development must stop. So there are many ways to overcome this valley of death, but as one approach, this is what we did. 3 years ago, we launched a new activity, which is SILA Foundation. This is like 100 people from SILA. SILA is a part of Kyoto University. They moved from SILA, from Kyoto University, to this newly formed foundation. It's a non-profit foundation. It's very small, but it's independent from Kyoto University. It's related to each other very, very closely. We rely on Kyoto University significantly, but nevertheless, it's independent. So SILA Foundation, with like 120 people now, has been working on various activities to overcome valley of death, such as GMP iPSL production, and also GMP differentiation of iPSLs to final products. Also quality control, including genome-wide analysis, and also like intellectual property license, these kind of activities. But the main function of this SILA Foundation is to provide GMP-grade iPSLs with affordable price. So there are 3 major iPSL lines provided or will be provided by SILA Foundation. There are 3 types. The first one is what I already described, HLA homozygous iPS cell stocks. So this is already existing. This iPS cell can cover up to 40% of all the Japanese population. But in addition to this, we are now working on genome-edited iPS cell lines. So we are modifying HLA genes in these HLA homozygous iPS cell lines to modify HLAs. So at least in theory, 10 lines would cover most world population. In addition, we have been working on autologous iPS cell approach. As I mentioned, at the moment it is too expensive and it is too much time-consuming, but by collaborating with many companies, we have been trying to reduce the cost and the time required for iPS cell generation significantly. So the reason why we work on all of these three different approaches is that we believe probably the best iPS cells will be different depending on each patient and depending on diseases. So we have to select. So that's why we do not focus on just one line, but we work on all three approaches. And we will provide these cell lines as affordable as possible. So as our president mentioned, Kyoto University has multiple on-site laboratories abroad. There are 3 in the States, and I have been involved in this iPS cell research center at Gladstone Institutes. So Gladstone Institutes is in San Francisco. Their mission is very clear: science overcoming diseases. And we have 5 institutes: cardiovascular disease, neurological disease, virology, genomic immunology, data science, and biotechnology. And in all of these institutes, iPS cell technologies have been widely used. And one of my colleagues, Kiichiro Tomoda, may talk a little bit about iPS cell research at Gladstone. And I myself have a lab at Gladstone, approximately like 10 or 12 people. And I have 2 major research activities at Gladstone. I'm still working on NAT1. I want to understand what NAT1 is doing, not only in ES or iPS cells, but also in many other cell types. So far, we have shown that NAT1 is also important in like hematopoietic stem cells and also small intestine stem cells. And also, with collaboration with Juan Carlos and other members of ARTOS, we have one project, small project, working on rejuvenation by partial reprogramming. So hopefully, through these activities, I hope I can be a small bridge between the States and Japan, between US and Kyoto University. So thank you so much for your kind attention. Thank you very much.
Thank you very much, Signa. We have time for a couple of questions.
I'm so curious about the cost of autologous therapy and whether you think the barrier, I'm curious about how much of a barrier the cost is for autologous therapy. Because as you know, I decided that's the way we were going to go. And I think we figured out how to keep the price down pretty well. So what do you think is going to be necessary for autologous, which is obviously the best possible therapy, to really be feasible?
So at the moment, it's very expensive. but we are hoping to reduce the cost up to like, let me calculate, $10,000 per patient. And hopefully we can produce like 1,000, we can produce iPSO lines from 1,000 people every month. So that's our goal by, for the next 2 or 3 years. And I think we can, it's easy to say, but I'm optimistic we should be able to do it.
I'm asking because most people say it's not possible. It's really good to hear you say it is.
Well, actually, Japan is very good at in reducing cost and time. We are a very good reputation in that. So I'm very confident.
Any other question?
Please.
It's coming, just 1
be
second.
patient. It's
1
coming.
second.
Well, thank you, Dr. Yamanaka. My question was, in terms of comparing the autologous iPSCs compared to genome edited from a donor, are there certain applications that you'd think the autologous would provide like a substantial benefit compared to the CRISPR, or does it seem like certain applications, the CRISPR edited cells would do just fine? Yeah.
So, like some patients suffering from blood disorders, they need to receive multiple blood transfusion. And if the patient has very, very rare blood types, not only like A, B, C, A, B, O blood type, but also like platelet type. After multiple platelet transfusion, you really have to match everything, HLA and HPA. But if his or her blood type is extremely rare, you cannot find donors at all. Or you can find only one donor in Japan or in US. And in that case, you need to rely on just one donor forever. And it's painful for both patients and that donor as well. So in that case, if you have to make iPS cells from that particular patient. So that may be very rare case, but I think there should be many other cases in which autologous transplantation is much better.
I feel we should go on. I was told that we should go on, so I would like to thank again Sinya for this exciting presentation. Thank you very much.
Thank you very much, Dr. Yamanaka and Dr. Belmonte. Now we are going to hear more about stem cells. Now I'd like to introduce Dr. Kiichi Tomoda to chair the session 1, Stem Cells and Cell Reprogramming. Dr. Tomoda, please.
Hi, everyone. My name is Kiichiro Tomoda, and thank you for the introduction. I'm a chair of this session, and sorry for Shinya, so I will not do any introduction of our program because of the time consuming. So, but let me jump to the session. So this session is focused on stem cells and cell reprogramming. And we have 2 leading scientists in the field. One, Dr. Belmonte, and the other, Dr. Takahashi. So let me introduce introduce you first speaker, Dr. Belmonte. He is currently director of Altos Lab Institute of Science at San Diego, and he is conducting extensively conducting the aging research there. So I believe he will share his latest research there. Please, Belmonte.
Thank you very much for your introduction. This first slide is just to illustrate our life trajectories, how from just these two cells, the oocyte and the spermatozoid, an embryo is generated, newborn human comes out. And for those of you who like soccer, you will have immediately recognized how this person, Pelé, at 16, 17 years old, it became really at the peak of his health. He's the only soccer player that has won a World Cup at the 17 years old. But like all of us, disease started to appear, and at a certain moment, he passed away a couple of months ago. Just want to illustrate this because he's one of the really best athletes in the world, and no matter how fit we are, at the end of the day, we are going to have disease and we are going to die. Now, in the last few years, and with that representation on the bottom left, we have started to map at the single cellular level the trajectory of many organisms. And you can see in this blue main trajectory, healthy cells that initially they are very well directed into one single trajectory, but as time passes, they deviate into other trajectories, which I want to illustrate then as disease cells. And the question I would like to address today is whether these disease trajectories can be converted into a healthy trajectory to maintain the life of organism longer free of disease. That's the main question that is interesting us at this moment. Now, this is a question whether cells can change trajectory that just about 70 years ago, still it was discussed by one of the pioneers in the field of developmental biology and epigenetics. And that was Conrad Waddington, that he proposed that cells, normally they don't change trajectory. They start from a pluripotent cell, they differentiate, but they don't go backwards. Now, the work of John Gordon and Shinya Yamanaka have demonstrated us that that's not the case, that an adult cell can be dedifferentiated backwards to a more plastic state. And this, as Shinya has mentioned, has led to many clinical breakthroughs in the clinical field, and he has shown today many examples that are today in the clinic of how to revert some disease states. Now, this experiment that was developed in the lab, the question is whether this is something that happens in nature. And here you have a few animals that have these extraordinary regenerative abilities. The axolotl, the zebrafish, the salamander here. You can see, for instance, the limb of the axolotl when it gets cut. In approximately 3 to 4 weeks, the limb will regenerate and will become an almost indistinguishable limb from the original one, with the same number of digits, the same pattern, etc. Different animals do this process in different ways, but one of the common ways, and this also happened, by the way, in mammals. So early on in mammalian development, You can see, for instance, the kidney. There are many other organs that do this as well, that they have this ability to regenerate. Here you see we cut one of the kidneys at about 1 day after the mouse is born. A few days later, you cannot clearly distinguish the one that you cut from the control one. So that's an ability that is present early on in mammals. It's just that with time, this ability diminishes. So I was saying that there are many different paths by which this regeneration takes place. But one of them, and perhaps the most common one, is the one I'm going to show you here, illustrating the case of the zebrafish heart. Like the axolotl limb, you can cut up to about 30% of the ventricular mass, and in about a month, the heart looks practically identical to the one amputated a month ago. And the question is, this regenerate here, where does it come from? Does it come from adult cardiomyocyte, or there is progenitors here in the remnant of the heart that differentiate and give rise to cardiomyocyte? The answer is just very quickly in these two pictures. This is the normal structure of a mature cardiomyocyte, and this is a cardiomyocyte that is undergoing now regeneration during the very early phases of the cutting. What you can see is that these sarcomeric structures in the normal cardiomyocyte are not that perfect in the undergoing regenerating cardiomyocyte, but they are still there. So if we look at the more molecular level details, what it seems to happen is that there is a small transient dedifferentiation of this cardiomyocyte, but immediately they start to redifferentiate, and together with increase in cell cycle gene expression, they take account for the regeneration of the cardiomyocyte and the entire heart. So the concept here is that cardiomyocytes in the zebrafish and in many other regenerative contexts, other cell types, they do not dedifferentiate backwards to progenitor cells, let alone iPS cells. They undergo this small transient dedifferentiation that, coupled with proliferation, allows the regeneration to take place in different organs and tissues. So that's a small summary here. So you can go from a differentiated cell to an iPS cell very early on, or you can partially dedifferentiate without losing cell identity. But the outcome is very different. While here you have a beneficial effect for the organism, if you were to do the 4 factors in a mouse and leave it for a long time, what you will get is losing of that cell identity, the appearance of tumors, and in the end of the organism. It's 2 very different outcomes. Now, if we look at what happened during a normal reprogramming experiment in the lab, you can clearly see that after adding the 4 Yamanaka factors, there are many cells that they take this trajectory that is not very well defined what it is, but they don't seem to go anywhere. And there is a few cells that undergo this path of making iPS cells, where we can distinguish several phases. There is a mid phase that we call, that varies for the species. In the mouse could be around 10 days after adding the factors, by which this is the point of no return. The cells have dedifferentiated, they are undergoing the path of iPS cells and they cannot come backwards. But very early on, in this early phase, there are many cells that don't take this path and they can come back to the differentiation stage, the adult stage. So the hypothesis that we put together, what is it? Is there any similarities between this early phase of transient dedifferentiation between regeneration and reprogramming? And could we then have this positive outcome that we see in natural regeneration by shortening the time of these 4 factors in the cells. The first experiment we did was in these mutant mice that they have an accelerated aging syndrome. So they normally live for 3 or 4 months because they have fast aging process. And the best treatment for this pulsatile or transient expression of the 4 factors we came up with was just to add it for a couple of days during the week. So 5 days, no 4 factors, 2 days expression of these 4 factors. And the result was really spectacular in the sense that this mouse is a very different mouse. It lives longer, has a better regeneration capacity. And when we look at the details of the different organs, you can see that they have rejuvenated. Now, if we were to leave these 7 days of 4 factors, these mice will get tumors and they will die. So what we think that is happening there is that we are just with 2 days, just trying to imitate what happens in these animals that regenerate, a transient dedifferentiation that somehow goes backwards and rejuvenate these cells. Now, this is just some details I don't have time to go through, but in this particular example, you can see how the cells of this progeric animal, they have a normal nuclear envelope that after the partial reprogramming becomes normal. They have an increase in DNA damage, which after the partial reprogramming becomes more or less normal. And there is upregulation of heterochromatin markers, which are key markers of this process of aging in most organisms. Now, this could be something that is very specific for this mutation, so it will be very important to do a similar experiment in wild-type mice. So we have done this, and we are undergoing a similar experiment in monkeys, and practically the results are identical to what happened in the progeria mouse. So with this data, we became very excited, and we decided to start a clinical trial. The clinical trial is just 1 person, and the volunteer is Sinya. And so we have started to add the 4 factors to Sinya, and you can start to see some beneficial effects in this slide here. So obviously, this is a joke, and the idea is, by chance, we initiated this experiment with an animal that has this fast aging process. But will this work in a disease situation that has nothing to do with aging? And for that, we generated a few animals where we can specifically deliver these 4 factors in a pulsatile manner in different cell types of the mouse. And I'm going to go quickly and tell you a few of the results that we are obtaining that in principle have nothing to do with the early phenotype of aging I showed you. This is a different mutation, nothing to do with progeria. This is the OB mice, a mutation in the leptin gene. On the left, you see the control mouse. On the right, you see the one that has been treated with this pulsatile 4-factor treatment. The fatty component tissue of this mouse gets shrink, and even though these mice don't stop eating because that's the problem of the mutation, they don't seem to become as fatty as the control one. So, but this is not only mutations. You can generate obesity by specific diet, in this case, in a high-fat diet. And you can see here that after giving this high-fat, to the mice, the wild-type mice, they become obese. But if you just give a couple of cycles, 2 weeks of this 4-factor expression, they start to reduce all the the fat component at the different parts of the animal. So it's not just a genetic link phenomenon, but it can be reproduced by the diet. There are other occasions that, for instance, toxins, high doses of paracetamol, of acetaminophen in this case, you know that they are not good for the liver. So if we give a high dose of acetaminophen to mice, in about a week, 100% of the mice will die. But if at the same time we just give a couple of cycles of this pulsatile expression of the 4 factors, about 50% of the mice survive, and this treatment is preventive of the toxic effect in the liver. And just to finish with the example, I could go on and on. This is something similar to the regeneration effect we saw in the salamander, in the zebrafish. Here we injure the muscle of a mouse, and you can see that after a couple of cycles of these 4 factors, there is more faster and increased regeneration as indicated by specific markers, in this case, here on the right of the treated one of the muscle lineage. Now, the question that we formulated at the beginning, is this an aging-specific phenomenon? I feel with these examples and many others, clearly is not. What we can say is that the pulsatile expression of the 4 factors in different mouse disease model help to alleviate and reverse the course of the disease. We're now extending these studies, as I said, to monkeys, a closer animal to us than the mouse. And the major area of focus for us is how these work. I have shown you several examples of diseases, either environmental, genetic, toxin, by which these factors, this pulsatile expression of the factors, can have a beneficial effect. It's very rare that a single medicine can do all these effects. This is something, I would say, difficult to believe, even for us. How does this work? And the concept I want to leave you today, I don't have time to go through the mechanism, is the following. The way that current medicine is practiced is trying to go very specific to the cause of the disease. It's an altered cell function that comes from another rare molecular function that in the end lies down in the malfunction of the gene. And what we are trying to do is either with traditional drugs or more recently through gene therapy, try to fix the problem at the very beginning. This is a highly disease-specific approach to treat disease. Now, if we take the example of the progeria, what we can do is do this gene editing, fix the problem there in this mouse, and then the mouse lives longer. But we can, at the same time, do this partial reprogramming phenomenon, and the phenotype is the same. The mice live for the same number of extra days, even though the mutation is not fixed. The mutation is still here, as well as the many other examples that I have indicated. And we know the majority of human diseases are not genetic mutations, single genetic mutations. After, as you can see in this graphic here, after 40, 45, there is a logarithmic increase in every type of disease, cancer, diabetes, liver disease, etc. And we cannot identify a particular gene mutation by which we can go and correct by gene therapy. I'll pass this. I see the light there. I don't have time. The concept that I want to leave you with today is the idea of this buffering and homeostasis system that probably all organisms have, which is, Shinya put the example today. He came from a plane from Japan, 12 hours of plane. He went to run, and he is here today. So probably, at least myself, but many of us will not be able to do this after a certain age. So the capacity, the buffering capacity that we have when we are young has nothing to do with the response that our cells have when we are old. So at an optimum stage, say when we are young, there is a high capacity to resist stress, perturbations in every cell of our body. And this maintenance of self-fidelity that happens when we are young can somehow overcome the appearance of the disease. I have shown you a graphic that after 40, 45, things start to go downhill. Many diseases start to come up. And probably what is happening there is that the curve now gets displaced. And then the stress tolerance, this buffering capacity that we have, is very much diminished. And the idea with these partial reprogramming protocols that we are developing is to try to increase somehow this buffering capacity. It's nothing specific. It's just trying to increase the fitness of the cell so that it can cope better with any disease, any perturbation that affects the health of the organism. Certainly it's not disease specific. And the way that probably it works, I don't have time to go through, is through modifying the epigenome. This is all what I wanted to tell you today, to introduce you to the hypothesis that in addition to the traditional approach of how diseases are treated today, which, by the way, in general, we don't cure the disease. We just ameliorate the symptoms of the disease, is the one that I have introduced with the possible avenue here, this partial reprogramming, that in many of the cases we have seen, at least in the mouse, it indeed reversed the disease, and probably by this buffering mechanism that endowed to the cells. And the most important slide of all is everyone in the lab in the last few years that have contributed this concept, and to the many collaborators out there. Thank you very much for your attention.
Thank you, Dr. Belmonte. I love your picture, senior. So we have time to have a very short question from the audiences. Any question?
So I was very impressed about your thought about the rejuvenation of disease state, which I think is important. But what happens in patients is that the disease state is kind of localized to certain areas of the body. So the question is it actually possible to target a particular disease site and try to reverse by different means to rejuvenate?
So this is what, in mice, we have been trying to do, to target this protocol to specific cell types where the disease originates. And the answer to your question in mice is affirmative. Now, as always, when we try to translate this to higher animals, I don't know the answer. But at least in mice, when we target specific cells that have that specific disease, the disease not only improved, but is even reversed.
Could nanotechnology's delivery capability be utilized for the approach?
One of the major problems always in this is the delivery of your cargo. How do you bring this to the cells, and how you control your cargo there, either with viruses, nanoparticles, and you mentioned nanotechnology. They are probably, this is one of the approaches that in the long term could help to target specific cells without the side effects that the use of viral vectors, for instance, have today. So the answer to your question, nanotechnology will be a major help in the application of this possible concept.
Thank you, Dr. Belmonte. So let me move to the next speaker. I'm pleased to introduce you Dr. Takahashi. He's a professor and our director at SIRA at Kyoto University. His team has conducted the first clinical trial for Parkinson's diseases using human iPS cell-derived cells. I believe he will share the update of his clinical trial related to talk. Please, Dr. Takahashi.
But sorry, I can't speak about the clinical trial outcome. It's secret. But to be honest, so I am not allowed to access the clinical data because I might give a pressure to the patients or doctors. So next year, maybe. So good afternoon. I'm Jun Takahashi from Kyoto University, SIRA, Center for iPS Cell Research and Application. And so first of all, I would like to thank all the organizers for this wonderful opportunity. And also, so I'm very happy that I'm back in San Diego because I spent 2 years in UCSD and Salk Institute at the Rusty Gage's lab. And also, I'm happy because I will talk about the stem cell biology and its clinical application, which I learned from Rusty. So the title is iPS cell-based therapy for Parkinson's disease. As you know, Parkinson's disease is caused by the progressive degeneration of dopaminergic neurons in the substantia nigra, midbrain, and it causes motor symptoms like akinesia, or rigidity, or tremors. Its main cause is the loss of dopaminergic neurons, and the worst thing is that it's progressive, so the patient is getting worse and worse. So in the first stage or early stage of the disease, the medical treatment can control the symptoms, but in the middle stage of the disease, the medical treatment alone cannot control the symptoms. That is the reason why we need to replace the lost dopaminergic neurons by cell transplantation. And iPS cells can proliferate forever, and also it can give rise to a variety of neuron cells, like neurons, muscle cells, or pancreatic cells, or liver cells, heart cells. And as Shinya Yamanaka told us, the clinical application of iPS cells disease to reveal the disease mechanism or drug development. But another type of clinical application is cell therapy. So we can induce the necessary cells to treat the disease, and we graft it into the body. So it's cell therapy or cell-based therapy. For example, to treat the Parkinson's disease, we need to replace the lost dopaminergic neurons. So we induce dopaminergic neurons from iPS cells and graft into the brain. And the grafted cells function as dopaminergic neurons in the brain. So it can replace these lost dopaminergic neurons and survive as long as possible, as the patient will die, maybe. So in this case, the grafted cells, the first can convert the L-dopa to dopamine. L-dopa is the main drug to treat Parkinson's disease, and the converted dopamine was transferred to the striatal neurons, and we can move smoothly. Or the grafted cells themselves can produce or storage or reuptake of dopamine. So that means we can expect the physiological supply of dopamine by the grafted cells. Okay. To achieve this, so we need to develop the protocol to induce dopaminergic neurons from iPS cells. So I don't have enough time to explain all of this, but briefly, we have developed feeder-free neuronal induction and also the purification of the midbrain dopaminergic neurons by sorting choline-positive cells. And we confirmed the safety and efficacy of these cells in Parkinson's disease mouse and rat. In addition, we performed the monkey study. So we used the cynomolgus monkeys, and we administered MPTP, which can kill the dopaminergic neurons selectively, and we can make the Parkinson's disease model monkey like a Parkinson's disease patient. And we grafted these cells into bilateral putamen and performed a long-term analysis, like a behavior analysis and brain imaging. And finally, after 2 years, we performed the histological analysis of the brain slices. This graph shows the change of the neurological findings, and the higher score means more severe deterioration. And this black line shows the control monkey without cell transplantation. And red line is the monkeys with Parkinson's disease patient-derived dopaminergic neurons. The blue line shows the monkey with healthy Brantier-derived dopaminergic neurons. In both two groups, the score gets less and less. So that means the neurological behavior or abnormal behavior improved in 1 year. So this improvement percentage is about 40%. To confirm the mode of action of the cells, we performed the PET study, 2 types of PET study. One is F-Doppler to detect the dopamine synthesis. Another one is PE2I, so to detect the reuptake of dopamine. So before surgery, the uptake of the very, very small because of we killed the dopaminergic neurons by MPTP. But after the cell transplantation, the uptake gradually increased like this. So red color means the higher reuptake of the ligand. So this means the grafted cells survive and function as dopaminergic neurons in the monkey brain. So after 2 years, we performed the immunostaining for tyrosine hydroxylase. This is the marker for mature dopaminergic neurons. So these are the images of the control monkey. So we killed the dopaminergic neurons by MPTP. So the TH staining is very, very faint. But in the case of the monkey with cell transplantation, we can see the dense staining here. So this is a higher magnification of this area. So you can see the cell body and also the neurite, the dense neurite here. So this is the axon of the dopaminergic neurons. And these axons extended into cover whole putamen like this. And also, so we counted the TH positive neurons, and we found that about 64,000 TH positive cells in one hemisphere, and totally 130,000 in average TH positive cells in monkey. So this is good enough to treat the Parkinson's disease patients. So with these data, so we thought we can move forward for the clinical application. So next, we started to evaluate the safety of the clinical grade final product. So this is a manufacture protocol from iPS cells to the final product. So in this process, we checked the quality in 3 points. The first one is the starting material of iPS cells. And the second point is during the sorting. And of course, the final, we checked the final products before shipping. So this table shows how we think about the quality check of the final product. So we divided the final products into cellular component and non-cellular component. and think about the safety and efficacy. The most important thing is what is the active cells in the final product. And also the non-target cells is also important. So we have to confirm that these non-target cells are eliminated from the final product. So for that, so we have a lot of experiments, and I don't have enough time, but the point is the active cells are dopaminergic progenitor cells, and we confirmed that the percentage of dopaminergic progenitor is more than 90% in the final product. Also, non-target cells are undifferentiated iPS cells or RA neural stem cells and transformed cells. So we confirmed that these cells are not contaminated in the final product by this method. And also for the transformed cells, we performed the genetic and also epigenetic analysis, and we confirmed that no dangerous mutation in the list of cancer-related genes here. And of course, the final product should be sterile and virus-free and no residual plasmid. And not only in the in vitro study, but also we performed in vivo toxicity and tumorigenicity and biodistribution test by using the immunosuppressed immunodeficient mice. So in this case, we grafted the most feasible maximum feasible dose into the striatum and observed as long as possible. So that means that we observed until the mice die, so lifelong span. So it turns out about 1 year, 52 weeks. So after 52 weeks, we confirmed the histology, and we found that no graft overgrowth or no malignant findings, and no metastasis to other organs, and also no graft-related toxicity. Okay, so by doing this, so we could confirm the safety and also the efficacy of the clinical grade final product. So then, the Japanese government and also the Kyoto University allowed us to start the clinical trial. So it was 2018. And our trial is allogenic transplantation of human iPS cell-derived dopaminergic progenitor cells. So this is the outline. So we took the blood sample from the healthy volunteer, as Asinya mentioned. So this is HLA homozygous healthy volunteer. And then we established the clinical grade iPS cells in the institute, and we induced the dopaminergic progenitor cells. So after purification and the stringent quality check, we transfer the final product to the Kyoto University Hospital again, and neurosurgeons transplant the cell to the patient. And neurologists and radiologists evaluate the safety and efficacy of the cells. Inclusion criteria, it is, of course, the Parkinson's disease patients, but the disease stages, the important. So in the early stages, as I mentioned, the clinical medical treatment can control the symptoms, so there's no need for cell transplantation. And in the severe stage, not only the dopaminergic neuronal system, but also other type of neurons, like cortical neurons or autonomy neurons. So it is too late to replace the dopaminergic neurons only. So the middle stage, it will be the target of our clinical trial or cell transplantation. The design, it is a phase 1 and 2 study at the single institute, Kyoto University Hospital. And it is a non-randomized open trial without control group. So we compare the status of the before transplantation and after transplantation. And the target number of the patient is 7. So we performed the 7 surgeries. So this is a surgical procedure. So I am a neurosurgeon originally, and so we have a lot of neurosurgeons in our colleagues, in our team. And so this is, so we perform the stereotactic surgery like this. So under general anesthesia, we inject the cells through bar holes here and into the bilateral stamen by using these long, thin needles. So we injected the 5 or 10 million cells per person. So we inject not the dissociated cells, but the cell aggregation called sphere, neuronal sphere. So its diameter is about 400 micrometer. And each sphere contains 10 to 20,000 cells. And we aspirate this cell aggregation into the needle and inject by hand like this. And per hemisphere, we use the 3 tracts. And in each tract, we inject 4 to 8 points like this. And 1 point contains about 200,000 cells. Follow-up period is 2 years. And primary endpoint is about the safety. So we will evaluate the adverse events like tumor formation, dyskinesia, hemorrhage, and so on. And secondary endpoint is related to the efficacy. So we will evaluate the neurological symptoms like UPDR score of time period and like Parkinson disease scores. And for more objective evaluation, we perform the PET study. So 3 types of PET study. One is F-Doppler to detect dopamine synthesis, as I mentioned in the monkey study. Another one is GE180. This is a ligand for the activated microglia. So we can detect the inflammation or immune responses in the brain. So we can monitor the immune responses after cell transplantation. Because this is allogeneic transplantation, and we will give the immunosuppressant drugs to the patient for 1 year. And after 1 year after we stopped this is the tacrolimus. So we need to monitor the immune response in the brain. So we use GE180 for this purpose. The third one is FLT, the fluoropyrimidine. This can detect the cell proliferation, dividing cells, so just in case. Okay, so this is the timetable. And before cell transplantation, we evaluated the neurological symptoms more than 6 months. And after surgery, so we evaluate the neurological symptoms and also brain imaging at 3, 6, 12, 18, and 24 months, like this. So now, so we have completed all the surgeries for the 7 patients. And so now we are in the observation period. And we will have the final outcome in the 2024. So it's next year. So I'm getting very nervous now. But anyway, so we have started and the clinical trial, and just waiting what happens. But we must go on further. So what is the next stages, or where are we going next? So I think there are several ways to go, but I think one of the ways to go is to combination with cell transplantation and other modalities. So I will show the 2 examples. One is cell transplantation and biomaterials. In this case, we used Parcan conjugated laminin fragment. So laminin fragment is like this and containing the active integrin binding site, so which binds to the cells. And another part is domain 1 of Parcan, so containing the laminin binding site and the heparan sulfate chains. The heparan sulfate chains can attract the growth factors like GDNF and BDNF, which are known to support the maturation or cell survival of the dopaminergic neurons. So we treated the dopaminergic progenitor cells with Palcan laminin before cell transplantation more than 1 hour, and then grafted into the striatum of the immunodeficient mice. After 2 weeks, we found that the number of the grafted or survived cells is not different from the control, but the number of Th-positive mature dopaminergic neurons increased after Palcan laminin treatment. So it means the laminin Palcan treatment can promote the maturation of the grafted dopaminergic progenitor cells. So briefly, the mechanism is like this. So the attracted the GDNF or BDNF stimulate the intracellular signals like RAS signals or PA3K signals. Especially the RAS signals can activate the ARC1, 2, and this promotes the maturation or neurite extension of the grafted cells. Okay, this is the first pattern of the combination. Another pattern is the cell transplantation and rehabilitation. In this case, we made the Parkinson disease rats, and we divided into 4 groups. One is control group. The second is treadmill training. It is a forced running on the machine, like this. And the third one is cell transplantation only, and the cell transplantation and treadmill training for 5 weeks. So after 6 weeks, we performed the immunostaining and found that the number of mature dopaminergic neurons increased by treadmill training compared to the cell transplantation only group. In addition, the neurite length from the graft is also promoted by the treadmill training. Okay, and when we examined the direction of the neurite extension, we found that only the dorsolateral direction is promoted, significantly promoted. So what is the dorsolateral striatum? So this is not our paper, but by others, but they say that the innervation from the cortex to the striatum, especially the trunk and the extremities, innervate to the dorsolateral striatum. So it is possible that forced running can stimulate this part, the dorsolateral striatum, and increase the neurotrophic factors or blood supply, which may attract the neurite extension from the graft. Okay. So we have a lot of treatment methods to treat the patients or diseases, like medical treatment, gene therapy, or medical devices and rehabilitation. But now we have ES cells or iPS cells to treat or to replace the lost cells by cell transplantation. So as I mentioned, so we can combine these therapies to enhance the function of the donor cells, like laminin Palcan, or also optimize the host environment, like rehabilitation. So I think these combinations will be the more important for the next generation cell-based therapy. So this is my message. Okay, so I would like to thank all the lab members, also the researchers in Kyoto University and outside the Kyoto University. And also for the clinical trial, I appreciate all the patients who contributed to the trial, and medical doctors and the co-medical staffs of Kyoto University Hospital. And also a lot of companies supported us, especially the Sumitomo Pharma. And financially, the Japanese government and IPSA Research Fund supported us. So I'd like to thank all of them in this opportunity. Thank you for your attention.
Thank you, Dr. Takahashi. So one question. So only we can have one question.
Okay.
Maybe.
Great talk. let's hope for the best in a year. My question is, have you had the chance to look at the mouse experiment, the epigenetic age of the grafted cells versus the host cells, especially in this rehabilitation training aspect, if by chance the improvement has to do with rejuvenation of these cells that you grafted, if you have had the chance to look at the epigenetic clock?
It's a very good point, but unfortunately, we didn't perform the epigenetic analysis.
Okay, one more question.
Oh, Jean. Hi.
Hi, Jean. Jean Loring, good to see you here.
Yeah, good to see you.
So I have lots of questions. The first question, of course, was how are the patients doing? And so you won't answer that. But I am really interested in, as you know, in autologous therapy, using autologous cells. So I'm interested in whether you see, maybe you don't know this yet, whether you see any deleterious or positive effects of immunosuppressing the patients after, because you leave them only for 1 year, and during that second year, is there any difference?
No immunosuppression. But I heard that the grafted cells survive.
The graft is still surviving.
Even with that, yes.
That's good. Well,
yes.
You do know something is going on.
Yeah. But I'm having I'm still want to do the autologous transplantation like you.
Yeah, I understand.
Yeah.
So do you see any downside about using autologous cells other than the cost? Is there anything wrong with using autologous cells if the costs were low enough? And that's the general question.
Yeah. I think it's all of you are thinking about the auto machine, culture method, and also so we need to next generation more effective iPS cell. It can downsize the cost. I hope, I believe.
Okay, thank you. Thank you again, Dr. Takahashi. So with this, I will conclude this session. Thank you for your attention.
Thank you to the speakers and Dr. Tomoda for chairing. We like to move to session 2. I'd like to introduce Dr. Fuyuhiko Tamanoi to chair the session 2, Quantum Science, Nanomedicine, and Radiation Therapy.
Okay, thank you very much. I'm Fuyu Tamanoi. I'm a professor at Kyoto University. I also hold cross-appointment professorship at UCLA. And so let me start here. So for the past several years, we have been having activity, meaning research meetings, discussions through online meetings and many other forms between Kyoto University and UCLA, and also with Department of Physics at UC Irvine. And the scientific topics or the theme of our activity concerns quantum beams such as X-ray or neutron beams, as well as nanomaterials, as well as radiation therapy, and finally tumor elimination and cell killing. So this involves the Kyoto and central part of California, Los Angeles, and Irvine. And 3 important discoveries have come out from this activity, and that's shown in the next slide. Could you go to the next slide? Yes. So the first discovery concerns something called OJ radiation therapy, which involves the use of OJ electrons, which have strong ability to cause double-strand DNA breaks, thus inducing apoptosis and tumor elimination. The second involves the radiotherapy application of high-density what did I put there? I'm sorry. The laser wake field acceleration, and the third one involves membrane signaling phospholipids and cell elimination. And today in this session, we're going to focus on the two right side topics. So I'll start with Professor Toshi Tajima, who's going to talk about the first topic. And Dr. Tajima is a Norman Rostocker professor, chair professor in the Department of Physics and Astronomy at UC Irvine. And I think without taking too much time, please go ahead and start your talk, Dr. Tajima.
Thank you very much for your invitation and introduction. I'm glad to report to you something that the previous session and this session are quite far away, so it may need to do lots of pictures instead of lots of equations. So next slide, please. Oh, do I have, oh, this one, okay, fine, thank you, I will do that. So here, I will talk about radiotherapy application. What the physicists do. I'm sorry, okay. What I'm going to talk about is radiotherapy application of what the physicists do, that is the accelerator. And usually when you think about accelerator is huge, at least modestly large. So if you go to the radiotherapy doctor, next door to the patient room, there is a room where the large accelerator sits, about 10 meters size. What I would like to do is reduce this 10-meter size accelerator to 10 microns. That is about 1 million times smaller. Okay, so that's our aim, and this is a realistic aim, actually. You learn in 20 minutes. Okay, so that's a radical jump. 6 orders of magnitude, 1 million times smaller. Okay, so how do we do that? Okay, so if we do that, by the way, then such an accelerator can be sitting on the tip of endoscopy. Endoscopy is the one that the surgeon will bring into your stomach or your butt, and then they cut the tumor and do something on that, right? So we can put such accelerator at the tip of endoscopy so that now I can do this laser wake field accelerator, do the job in 1 million times smaller than the accelerator you may have seen. How do we do that? Well, what I would like to tell you is I have invented something called wake field. Wake field is like a motorboat. Behind the motorboat, there's a wake. So this is the wake field. I would tell you through the pictures instead of equations, hopefully. And this can originally intended to make a high energy accelerator for physicist purpose. But now I'm turning that to the medical people's purpose, low energy accelerator in a small size accelerator. And how to do it? We can use a fiber laser. Fiber laser is very small and tiny, and you can put it inside your stomach or in your butt. And therefore, it becomes endoscopic radiotherapy. That's the current. From the invention of this acceleration method about 45 years ago to the current status. Okay, how do I? Okay. All right, so this is the wake field. Mother duck is causing the wave of the ocean ripple like this. And these ducks, the baby ducks are lazy enough that they want to ride on the waves that generated mother. So it's a ride on it. So that's called the wake field acceleration. So duck is making wakes, and the ducks are baby ducks are being accelerated by the mother duck wake. wake by a duck. And in other words, the nature or mother duck showed us how to accelerate effectively. So a little bit more real to our medicine, but before that, this is the same thing. A duck is replaced by motorboat, and the motorboat is making a wake field behind it. Now I'm going to do the laser pulse instead of a motorboat. This can be micron size pulse, and behind the micron size pulse, you can generate a wake field of the micron size. So this is what we would like to do for the purpose applied to the medicine. You can see the big waves like this, or tsunami waves, but now we are making very tiny micron size waves in your stomach or other parts of the body. So simple like that, right? Well, not so simple in terms of equations, so I'm not going to go into the equations, but this is what we invented in 1979. The formula is relatively simple, and the experiments done by hundreds of labs in the world, around the world, up to even the tens of GeV or near TeV, depending on the choice of the plasma density. And if you go up the density higher, energy is lower, but it's getting smaller and smaller. That's what I would like to do. And then we call the near critical high density acceleration of the wake field. That's what I recently invented. So this is about 20 years ago. People have now demonstrated this idea into the actual lab. First experimental demonstration of this accelerator. Laser looked like this at the time, which is about a few meters size. And then eventually they got this small accelerator, few centimeter size accelerator, generating 4 GeV electrons, whereas the conventional accelerator takes hundreds of meters to reach 3 GeV. So nature got excited. Nature, in this case, the journal, not the nature, Mother Nature, but Nature. But journal excited, wow, dream beam. So that's what they published in 2004. Okay, again, a little bit the physics work is necessary. We did a lot. My student, Dr. Nix now, he has published how to do this in non-relativistic regime in near critical density, which makes the micron size accelerator. These are lots of works he has done, and we designed where the good parameter, soft spot, and how to copy the mother duck success story inside your micron accelerator stomach. Again, another doctorate candidate work. I have no time to go into the detail of these works, but we know by these series of computer simulation where is this right spot resonating electrons get the tiny acceleration, a nice beam, etc. And the quantitative measurement of this thing can be done by this study. Again, I will skip for the benefit of the time. And this is my latest student's work, Mr. Barraza. Strickland is probably famous to you. She is the third Nobel laureate, woman Nobel laureate in physics. She is working kindly with us. And during this work, we identified the spot where the good thing happened. It's a very efficient spot, near critical density. 1 it means a critical density, slightly less than 1, where we can generate a very tiny micron scale accelerator. And this is how you get tens to 100 of KV type of electrons over 10 micron size. 10 micron size. You can't even see the size of the accelerator. So this is the kind of guidance by our theoretical and computational work. And this is what we try to design to make actual experiment of this one. And we did recently, which is just submitted to, well, not yet even submitted, but to be submitted to a journal on that experimental demonstration that we have done recently. The results here are not here yet, to show. So this is the one we recently carried out And what do we do to make accelerator smaller? Not just this idea of a mother duck is pulling the wake field. We want to identify small specimen so that this specimen can generate micron size acceleration. So this is the nano fibers, carbon nanofiber. It's called CNT, carbon nanotubes. So size of this is 1 micron, so that each carbon nanotube is about 10 to 100 nanometers diameter. Length is several microns, few microns. And this is a forest of nanotubes. I call it carbon nanoforest. So this is the material which can be generated by current nanotechnology very easily, or relatively easily, I should say. And this is my target. Okay, how do I do this target? Well, okay, here like a typical carbon nanotube, which is sparse like this, and then you inject the micron size laser pulse into it. Then this micron size laser will accelerate the electron forward over probably few micron. Few micron is so tiny that you can't even see it in your eye very well. So it is a very tiny size of accelerator. Literally can be put on tip of your endoscopy. So that's what we can imagine, and we are taking this kind of material. All right, so we have been again using some kind of laser, which is we call the fiber laser. Fiber laser is not big laser as I showed in the previous picture, but now it is just a line of fiber, few millimeter thickness, and then it can be long, and you can put it fiber into your stomach or in your butt, or even in the pin into your body if it's necessary. So using this type of parameters of the fiber laser, we have generated where and how much, and about typically 2 micron size target, just as you show before previous page on the carbon nanotube over micron size. So this is the latest work we have identified. And what we do is that this is the typical laser, high power laser called CPA laser, from which Professor Gerard Mourou and Dana Strickland got Nobel Prize in 2018. So this is the typical laser that the current world lab, 100 labs are employing this kind of laser, but now replacing this kind of relatively large laser, this is about a few meter size laser instead of hundreds of meters size, to the fiber laser. This is the fiber, laser fiber, and then these fiber can be insertable to your body, like this here, or even into your other parts of the cave. All right, so this is the fiber laser. And then again, this is too complicated to go through the entire numbers, but the point is that Dr. Shah has studied what kind of laser is probably most suitable and employable immediately under the current technologies, and this is the parameters he identified, and the intensity identified. Again, Professor Dana Strickland and we are employing such parameters together to carry out the experiment at her lab, and the fiber laser look like this, very tiny shape of the, just like your hair type of thing. And in that kind of string, the laser can be amplified and propagate into your body and the spot. So this is the typical laser, fiber laser. So that's the one we would like to employ. In contrast, this is what the current radiotherapy therapist employs. Probably some of you may have gone to the radiotherapy hospital. This is Professor Dante Rohr at UC Irvine Hospital. He is using such an injector of electrons. This is the one electron comes in and that comes out. It can be also X-rays, can come out, and the patient is sitting here and irradiated. In this kind of irradiation, as you know, the radiotherapy means that the radiation, X-rays, neutrons, or electrons are generated outside of your body, of course. In this case, next door, there's a big room here, and here, of course, accelerator sits. Accelerator size is about 5 to 10 meters next room. And patient does not see the accelerator. And this accelerator put the electrons in or X-rays coming in, and then they shine the patient here. That means that the current conventional radiotherapy means that the radiation is outside of your body and they shine on your body. And then, of course, by definition, cancer is inside of your body. So you have to penetrate your body until you reach to the cancer. So that means that, let's say inside of my stomach, I have a cancer. Then I have to insert electron, let's say, and the electron energy has to be like MeV to penetrate, let's say, 5, 10 centimeters, because it's slowed down until it reach to the cancer. Until reach the cancer, what the electrons does, of course, the electron will kill your skin cells, as well as your fat cells, as well as stomach cells and your body bone cells, and then finally reach to the cancer cells and let's kill the cancer cell. So in other words, there is a collateral damages done by these electrons or X-rays until you reach to the actual target of a cancer. Now, what we want to do is instead of irradiate your radiation from outside to the inside of your patient, we want to generate 10 micron size electron accelerator inside of your body so that you don't cross the surface skin to the body and finally reach skin, the cancer, but rather, for example, if I have a cancer in my stomach, surgeon will insert a fiber laser endoscopy, and he will see through endoscopy, oh, here's your cancer cell, and he will cut this cancer tumor. Now, if he's lucky, cancer is gone by the scissor incision of this cut of endoscopy. But you never know that the apparent lump is only the cancer, but there might be spread cells which you can't see by the eye. Therefore, we want to do is shine that part by these electrons, shine inside your stomach, size of the accelerator 10 micron. In that case, energy of electrons do not need MeV because you don't have to penetrate the 10 centimeter, but rather only size of the cancer cell, which is a few microns. Therefore, you need only 10 kV to only up to 100 kV. You don't need to go MeV type of accelerator. So that's why another advantage of making small and efficient and light damage accelerator and radiation. Okay, so that's the idea. It doesn't move. Okay, yeah. In addition to the accelerator itself, usual radiotherapy comes with a lot of applicators such as this, several centimeters of all kinds of hardwares. You kind of put this way and that way in this. If you have a various organ, depending on organ, this kind of thing. Now, endoscopy, we don't need this because it just comes in front of the cancer cells. Now, this is the schematic diagram, how it would look like, our tip of endoscopy fiber accelerator. So the fiber comes in like this, and this fiber laser will shine on this carbon nanotube nanoforest I showed earlier. So this is the nanomaterial, nanocarbon fibers. And then the electrons go out over 10 micron, and then this is the target cells So this is the kind of a compactified accelerator and a compactified radiation therapy gadget, few tens of micron size, and accurate because you are now, surgeon is looking through the endoscopy where you cut such an area, and the cheap we will see in next slide. This is the provisional estimate by Professor Rohr. He compared this laser wake field accelerator-based endoscopy versus other radiation therapy cost, roughly estimate, maybe like a factor 10 cheaper, according to his estimate. One more last slide is combination of this tiny accelerator in front of the cancer cells. On top of that, even further make efficient and targeted therapy to be realized. Professor Tamanoi invented this vector medicine, nanomedicine, which can seek and attach to the cancer DNA like this. This is iodine. So if electrons are generated in front of this cancer cell by our micron size accelerator, electron come through it like this. But if electrons see the iodine, hides the material, electrons decelerate right around here, and then of course right around here is a cancer DNA, and the DNA get destroyed. So it is preferentially destroying the cancer DNA. On top of, you see in front of where you want it to shine, and it don't penetrate your body. So this is the idea. And so in conclusion, by choosing appropriate parameters such as critical density nanomaterial, we can generate more than 10 kV but less than MeV electrons through fiber laser. Fiber laser technology now has been sitting, waiting for us to be deployed, and then using that we can make endoscopic electron accelerator radiotherapy, and furthermore, nanomolecule vector can further target specific microscopic cells of the cancer or DNA itself. So thank you very much for your attention.
Thank you very much, Dr. Tajima. We can take a question here at the back.
So there is a German company, Storz, that is located near you that sells a endoscope for mice. And at Kyoto University, Mark Taketo has mice that generate tumors. Have you considered an animal model for this?
Well, we are far behind that front yet. Okay, so we are still fast approaching this kind of conceptual design of the accelerator. What we did last fall and until now, and as I mentioned that we are preparing the paper to publish, is the still bigger laser, not the tiny fiber laser yet, but the conceptual experimental demonstration at this moment. So in other words, I have 2 steps to go. We have to now employ fiber laser to make a compactified one, then they go to your step, all right, to do that kind of thing. So we are still 2 steps to go, but I'd like to hear your recommendation.
Well, you're just in time to start your IACUC protocol.
Yeah, great. Yeah. So I'd like to ask
that's a good
your
suggestion.
recommended factory or groups. Okay.
Yes, that sounds good. Other questions? Why carbon nanotube? Did you say anything about the advantage?
Okay, carbon nanotube is very designable in the arbitrary, well, not arbitrary, designed diameter. Such as if I want to care about 100 nanometers, you make a silicon substrate with certain shape, and then blow off on the carbon air, then it will pile up this way. So it will designable to make 100, let's say, nanometer diameter. Then, of course, the thickness of the carbon nanotube is angstrom. That means that if you make 100 nanometer diameter carbon nanotube, then we know the density of electron is 100 nano, that is 4 orders magnitude. So 10 to the 23 minus 4 is at 19, for example. So you can design the density specific to our desired way. So that's why I take that.
Thank you. So it's a
There are
design
other reasons, but it's too complicated to go in every reason for that.
Well, let's thank Dr. Tajima again.
Thank you.
So the next speaker is Dr. Jun Suzuki, and he's a professor at the Kyoto University, and he's deputy director of ICEMS, which is an Institute for Integrated Cell Material Sciences at Kyoto University. And he also established an on-site lab with Academia Sinica in Taiwan. So please go ahead, Dr. Suzuki.
Thank you for your kind introduction. And I hope that what we are doing is a fundamental, but you can enjoy our science, but also our approaches, which might be applicable to your science as well. So I guess everyone is interested in the cells, but in a different way. How I see cells is like cells establishing asymmetrical distribution of ions across membranes. For example, like sodium and calcium is exported from to the extracellular side, but when cells are activated, sodium and calcium are coming to be activated. So this is a fundamental in how neurons are activated. And then when these molecules were identified, the researcher progressed. PS1 was identified in San Francisco, and TRPV1 is in San Francisco, and PS1 is found in San Diego, I guess. But not only in ions, but also lipids are asymmetrically distributed. Here I called a phosphatidylserine. This is not a pluripotent stem cell. Okay, phosphatidylserine, PS, I call it. So PS normally locates on the inner side of the membrane, but it's exported on the cell surface when cells are activated. But in contrast to the ions, not much known about the lipid asymmetry. So that's the reason why I try to open this black box. And then how lipid asymmetry is maintained is usually using energy. Lipase try to maintain at the inner side of the membrane. But when cells are activated, scramblers are activated, which transport lipids bidirectionally and non-specifically. But especially about scramblers, no one knows its molecular identity. So when I was a postdoc, I started to identify it. But some people say it may not exist. So actually I was nervous at the time. And it took 6 years in total, but finally I came up with 2 scramblers. Okay. Oh, it's not moving, so I move here. So one is XKR8, which is activated by caspase-mediated cleavage. And when PS is exposed in dying cells, that can be recognized by phagocytes and engulfed by phagocytes. And then also, fortunately, I could identify another scramblase called TMM16 that is activated by intracellular calcium increase. And when platelets expose PS, coagulation factors in bloodstream binds to PS to activate and to stop bleeding. And if this protein is deficient, Scott syndrome, bleeding disorder occurs. And then when I became independent, I more focused on the neurons. Because in the case of neurons, not only in the dead cells, but a part of living cells can be engulfed by phagocytes. Because in the case of a neuron, you need to use the same cell for life. So unwanted part or aged part expose PS and to be engulfed by phagocytes. But its molecular identity is completely unknown. So I started to work on it. And then I focused on the one molecule called XKR4, which we identified. And that is cleaved by caspase and activate, but cleavage is not enough. Another mechanism is there. So I try to identify the molecule. And also, when the XKR4 is not there, what happens? These two, I got the questions. And as for a physiological role, just in the one slide I want to introduce. When XKR4 is missing, it shows hyperactivity and attention deficit, and also shrinkage in the brain region called prefrontal cortex. And also, XKR4 SNP was found in ADHD, attention deficit hyperactivity disorder patient. So this is more relevant to the human model of ADHD. That's what we believe. But today, I want to focus more on activation mechanism of XKR4. So after cleavage, XKR4 forms a dimer, but this is not sufficient to be activated. Under the control of caspase, XKR activator needs to be activated to activate XKR4 dimer. But as you know, once caspase is activated, death occurs. So cells cannot proliferate. It means you cannot enrich the cells which you are interested in, and all information has gone. Our challenge is to identify the factors from dying cells. And then we use the CRISPR short guideline library. And this is the most important slide in my talk. And we first try to use the neurons for the screening, but it's very difficult. It's heterogeneity is there. So I try to reconstruct XKR4 into lymphocytes, but still XKR4 is activated. So we express XKR4 into a lymphoma and induce a CRISPR short guideline library by lentivirus, then induce cell death. And then non-repeat scrambling cells are sorted by flow cytometry. But there are two problems here. Once caspase is activated, DNA is fragmented. So all information has gone, disappeared. So one trick is I first try to make a DNA knockout. So then now even cells are dead, DNA is intact. But still cells cannot proliferate. So you cannot enrich important information. So then what we did is we collected genomic DNA and amplified the short guideline region by PCR and reconstituted into a lentiviral vector, and then use this library into new cells and perform next screening. So using this procedure, even using dying cells, you can enrich important short guide RNAs. And many people ask me, just one-time sorting, you can identify the factors, but actually not. Just one-time sorting, mostly junk. So you need to repeat these processes. And I want to show raw data. And we call this screening as a revival screening. And after an apoptotic stimuli, we sorted 1% of negative population and constructed next library around for 3 weeks. And we introduced it in the cells, and still enrichment does not occur. But we sorted and constructed library and introduced in the cells. And now 1% became 2%. So you may doubt, but we are pretty much confirmed. Probably enrichment may occur. And after next sorting, now it became 20%. So now that's enough to perform next generation sequencing analysis. So 6 short guide RNAs are designed per one gene, and we consider more than 3 should be the true. And then total, based on the total list, we ranked it from 1 to 20. And we found that cytochrome C and alpha F1, it's upstream of caspase 3 is already identified. And probably among these 18 genes, true hits are there. And then how are you going to do? So actually, you try 18 all genes by short guide RNA. Actually, that's what my student did. But actually, we thought it's a better way to narrow down. So to narrow down the caspase 3 downstream. So this is a point. Because we already enriched as a library, we introduced into cells and sorted repeat scrambling negative cells and fixed with PFA and detected by caspase-3 antibody, and then we found that only 5% is active for the caspase-3. So if we sort this region, probably the factors can be enriched. And it was like that. So previously, 8th ranked gene became top after this sorting. And then what the gene was, actually XRCC4, DNA repair complex protein. To be honest, we are so disappointed because we wanted to identify factors which activate plasma membrane protein, which should localize to cytoplasm, but we identified a nucleus protein. So disappointing. But we already got one gene, so we need to do it. And then when XRCC4 is knocked out, caspase-3 is normally activated, so it should be downstream of caspase-3. Then we asked, it's cleaved by caspase? And yes, it's having a caspase cleavage site. And after apoptotic stimuli, XRCC4 is cleaved. And then when the 2-aspartate was changed to an alanine, it's not cleaved anymore. So what we identify by screening was a caspase-3 substrate. Then we became to be interested in this gene. And this is the most important. When XRCC4 is knocked out, XKR4 on plasma membrane has no activity anymore. So it means XRCC4 in the nucleus is critical to regulate plasma membrane proteins. And when we introduced wild type, the activity was recovered, but caspase resistant mutant was introduced, it was not activated. So XRCC4 needs to be cleaved by caspases. Then we asked its localization. So many researchers in the world say like it's localized in the nucleus. I doubt it. So I tried to see its localization by using fusion protein, but it's mostly localized in the nucleus. But in the case of XKR4, it's plasma membrane. How these two proteins can meet each other? This is our next question. And then when fluorescent proteins conjugate to the N-terminus, and after apoptotic stimuli, still N-terminus is localized to the nucleus, but when it's added to the C-terminus, this region is coming to the cytoplasm. So now, this XRCC4 C-terminus can meet with XKR4. So, and this C-terminus doesn't have any like structure. It's intrinsically disordered region. So it's my liquid-liquid phase separation may occur, and then to activate it. Then it's a very simple experiment. So then which is important? It's probably C-terminus. We examine it. So when we introduce N-terminus, no activity. And when we introduce C-terminus, actually it's almost no activity. Why? Because we need methionine, but after a caspase cleavage, its solution is exposed. So we just added 10 amino acid and just activated, just only a C-terminus can activate XKR4 on plasma membrane. So to confirm this result more, we produce only C-terminus in E. coli and electroporated into the cells. In that case, no caspase activation, no cell death. Living cells keep on exposing PS all the time up to 6 hours. So now we came to find out DNA repair proteins fragment C-terminus can activate plasma membrane protein XKR4. So I'll summarize here in the model. XKR4 stays as a monomer. After caspase-mediated cleavage, it forms a dimer to be activated. But this is not sufficient for activation. XRCC4 in nucleus also need to be cleaved by caspase. Then C-terminal part is released into the cytoplasm, and that directly binds XKR4 and activates on it. So we think this is a new concept that the nuclear protein fragment can regulate plasma membrane protein. And also, once XRCC4 is cleaved, its function becomes null. So people believed that this cleavage is to make XRCC4 non-functional. But that fragment also has a function. This is, I think, new discovery. So then using this revival screening, we recently identified a new scramblers. It's actually 2 more. But last 5 minutes of my talk, I just want to introduce how our approach can introduce it into a biomedical field. So now using CRISPR screening, especially revival screening, now you can sort your phenotype-based sorting, and you reconstruct the library. And if you can repeat on it, you can narrow down the factors. So now we are using dying cells, but also growth-arrested cells. We can enrich critical factors. Cells do not need to proliferate. Even using primary cells, it works. But many people ask, using cell lines, it's relevant to vivo? But that's also we can analyze, because now we enrich as a library. This library can be injected in a mice directly, and we can examine whether the factors are relevant to in vivo situation or not. So one example is like one student, PhD student, and she's actually a chemist. And when she was a master course student, she designed a probe which can detect triacylglycerol-rich lipid droplet. And we are not working on a lipid droplet, lipid asymmetry, but she came to my lab and she just want to use these probes to know how NASH occurs. NASH is like in a hepatocyte, like lipid droplets is actually accumulated in a TAG-rich, not cholesterol ester-rich. So she wants to know the mechanism, how these are regulated. She has a probe, and now we have a revival screening, and combining these 2 strategies, now we could enrich the library after a 4th time sorting. And now this enriched library is directly injected into a mice liver, and then we examine which factors are relevant to in vivo situation as well. So from cell lines, also in vivo is also quite seamless. That's what we believe. And also another approach, what we are currently doing, is in a case of, most cases we use cultured cells, but in other cases, we directly inject the lentiviral library into mice tissues, and then perform sorting and repeat on it. So it's more like in vivo situation, revival screening is possible. So one student, Yuki Noguchi, now he's a PhD candidate, also joined my lab 3 years before, and he said he's interested in a sperm PS exposure. When sperm undergo capacitation, they become fused with oocyte. But how PS is exposed in head of sperm, no one knows. Because in the case of sperm, cell lines, you cannot use cell lines. So bicarbonate and calcium is incorporated and expose PS, but we don't know what happens here. So he said he wants to use this revival screening technique to reveal these processes. So what he did, using a lentiviral library, introducing into a testis directory, and then germ cells are infected by a lentiviral library, and after a capacitation stimulation, based on a calcium signaling, he sorted capacitation negative cells by flow cytometry. But in the case of vivo situation, cell numbers are limited. So it's quite hard to perform PCR later. So in the case of vivo, we perform 4 genome amplification at once, then apply to a PCR to amplify the integrated short guide RNA, and then introduce into the next mice. So from germ cells introduction to performance, screening is for development. It takes 2 months. So it's like each phase is about 3 months, 3 months. So then he actually performed twice. And at the first time sorting, he obtained 45 genes. And then he used this library and injected in another mice. And 16 weeks later, he narrowed down to 20 genes. And among these 20 genes, he just started to analyze top 7 genes and found that RD3 makes cells deficient in calcium influx. So at least RD3 is important to regulate sperm functions. So not only using cell lines, but also using in vivo tissues, we can start to identify the factors. And we think that this approach can be applicable to any kind of disease condition. And then in cell lines and mouse tissue, we can elucidate the factors which might moderate disease condition. That's what we believe. Okay, I want to finish my talk. So first, my talk was done by Masahiro Maruoka, my first staff postdocs, and also Yuki Noguchi, PhD student, and also Pan Chan, PhD student. They work together, and they are going to having PhD soon. And then now we have 7 nationalities, and one of the purpose of this on-site lab is to increase diversity as well. So now we have 7, and then we are going to have 3 more. So among such a diversity environment, students have different ideas and way of thinking, so that make our research accelerate, that we will believe. Okay, thank you very much.
Thank you very much. I think we can take questions. Any questions? There was one slide where you talked about cleavage in the nucleus and then the movement of the C-terminal fragment going to the plasma membrane. What is the mechanism for that?
Yes, so actually it has been known that many DNA repair proteins, such as BRCA1 and also RAD50, they are all cleaved by caspases. So people believe that caspases enter into the nucleus and cleave these proteins to make it non-functional. But the people's idea stopped there. But now we came to find out that cleaved fragment also has another function. When it releases into the plasma membrane, because it's a small fragment, probably it's just diffused into the cytoplasm. Then meet with exhalation from the plasma membrane to activate it. So this is what we believe currently, how the exhalation fragment can activate exhalation from plasma membrane.
I see. Okay. All right, thank you. Yes, go ahead.
Thank you very much for your impressive talk. I have a question about the sperm antiviral covalent screen experiment. And in the first round, 45 genes, and the second round, 20 genes are mostly overlapped or not?
Yes, like among 45 genes, like it became about half. But actually, in our first attempt for the revival screening in testis is not good, to be honest. Because in the case of germ cells, once cleavage occurs, cells easily die. So we lose many kind of short-guided introduced germ cells. So then kind of coverage itself is not good. So now we are performing it in liver cells. It may work well, but in the case of germ cells, only 45 genes we could obtain. But among these, about half enriched. And the top 7 genes we start to analyze, and we identify the factors which is involved in spermatogenesis. Is that okay?
Yeah, thank you very much.
Okay, thank you.
Okay, thank you very much. I think as you can see, there are many topics discussed in this session as well as before. So hopefully in the future we can have a lot more in-depth discussion, and let's thank the speakers for the session too.
while you're taking seat, I realize I forgot to introduce myself. I'm Kaoru Kitajima, and I'm a professor and the vice director of the Kyoto University's International Strategy Office, and I'm in charge of the North American networking of Kyoto University. We have a Kyoto University North American Center and 3 on-site labs, as you were told earlier. And now, while you're taking seat, in the interest of time, I'd like to start introducing the session 3. And session 3 will be chaired by Dr. Silvio Gutkin, who is a distinguished professor and chair of the Department of Pharmacology, School of Medicine at the UC San Diego right here. And he's recognized for his research in the development of precision cancer therapies, multimodal immunotherapies, etc. Dr. Gutkin, please.
So thank you so much. Let me, here's that. Dr. Kaoru Kitajima. So thank you so much. So I have to tell you, so this is amazing to see all of you here after several years of interacting with my colleagues and friends in Kyoto University. And certainly, you may remember last year organizing together with Masa here, the series of lectures from cancer immunotherapy with speakers from UCSD and Kyoto University. This is just a dream come true to see all of you here, and especially being part of the immune therapy session. So let me, I will just introduce, I would like to invite our first speaker, is Nagashiro Minato. Dr. Minato does not need introduction. He is certainly the president of the Kyoto University. He has made major breakthroughs in cancer immunotherapy, in immunotherapy in general. And certainly he revolutionized altogether cancer therapy when he identified the opportunity of using targeting anti-PD-1, anti-PD-L1 as an immune checkpoint blockade for cancer therapy. That led, we all know, Dr. Honjo's Nobel Prize recognizing these discoveries, and history was made. This time, he will not be talking about cancer immunotherapies. His talk will be at the intersection between aging and immunity, and we look forward to your presentation. Thank you.
Okay, thank you for the nice introduction. And I wasn't expecting talking in this scientific session other than opening remarks, but and also today I'm not going to be talking about cancer immunotherapy, so just please forgive me. Okay. As you may know, Japanese are blessed with probably the longest average lifespan in the world. And still the major cause of death of the aged is cancer, amounting up to nearly 50%. So remaining to be a major threat, major challenge of life of the aged. Nonetheless, life and death is not the sole issue of the aged. And yeah, here's the word by Simone de Beauvoir. I'm not a French speaker, so I'm not sure my pronunciation is correct or not. But anyway, the Beauvoir once said in her popular book that we rarely encounter the plain or pure state of the old age. The aged almost always suffer from many chronic diseases. And this graph in Japan actually indicates that the major common chronic diseases tend to develop at about ages, say, 60 to 70, almost synchronously, implying that aging per se might underlie the development of these very common chronic human diseases at ages. Although those common diseases not be immediately life-threatening, but they certainly affect the QOL of the aged. So today I would like to focus on the aging of immunity and its possible involvement in such age-associated common chronic diseases. Although it's not that easy to define the aging of immunity, but it has been known that there are significant alterations in the compositions and function of immune system with age. Typically, we and others have reported that unique cell populations that are rarely encountered in the young are developed and accumulated with age in both T cell and B cell compartments. For instance, senescence-associated T cells or SAT cells show quite unique phenotype and characteristic features of cell senescence. And functionally, those SAT cells are refractory to optimal T cell receptor antigen stimulation, but have a strong potential actually, for abundantly secreting pro-inflammatory factors or cytokines, chemokines, and so on and so forth, which may be reminiscent of so-called senescence-associated secretory phenotype, which often called SASP. Similarly, ABCs, this is age-associated B cells, show the phenotypes quite different from regular B cells, the majority of B cells in the younger. They also show vigorous responsiveness to toll-like receptor 7 in response to single-stranded RNA, such as viruses, as well as a potent antigen-presenting capacity. Notably, these particular B cell subsets, which increases only in aged population, has intrinsic predisposition for autoantibody production, which I come back later. And as you know, for optimal immune response to take place, the typical interaction or collaboration between T cells and B cells required. As you know, T lymphocytes and B lymphocytes represent the major two subpopulations of the immune system, and they develop very distinct manner. For instance, T cells are developed exclusively in the organ thymus with aid of thymic epithelial cells, whereas B cells are developed in the bone marrow, both from the same hematopoietic progenitors. And regularly, it's all in the text, optimal T cell B cell interactions are required for the production of antibody against foreign antigens. And in response, in response to those foreign antigens, T cells and B cells, I mean specific T cells and B cells, show robust clonal expansion, followed by the differentiation into functional effector cells to exhibit optimal immune responses. But importantly, those are typical TB interactions in the immune system. But importantly, thymus somehow shows early involution with age, which becomes already evident in adolescence. So thymic supply of naive T cells steadily and progressively reduced or declined with age. So to compensate the reduction of the supply of T cells, naive T cells from the thymus, now peripheral T cells begin to divide autonomously, even in the absence of any specific antigen. And the process is called homeostatic T cell proliferation. And this involves, again, T cell B cell interaction. But in this case, T cells show the self-antigen on regular B cells. And you have to remember that T cells have been selected in the thymus during the development. And this selection is based upon the self-reactivity. That means all the T cell receptors expressed on the peripheral mature T cells have intrinsic self-reactivity, although the affinity of the receptor is normally very low. But this is a very unique feature of immune system. Taking these advantages in adult ages, since T cell numbers otherwise is going to go down progressively because thymus is progressively in shrinkage. So those T cells now divide by looking at the self-antigens expressed on normal B cells. But this is only to sustain the number of peripheral T cells, not to cause immune response. So the response should be very much limited, enough to induce proliferation, but not activation to cause immune responses. So this is very unique pattern of TB interaction, where you have very low affinity of receptor, antigen, or ligand interactions, enough to induce slow rate, non-clonal proliferation without causing any active immune responses. So this is very different aspect of TB interaction, but it occurs consistently in adult stages. And to avoid still possible activation of the immune responses, the PD-1 and PD-L1 system made a very crucial role in this homeostatic T cell proliferation. So that's the second aspect of TB interaction. Then there's another T cell B cell interactions. And as you might imagine, this homeostatic proliferation occurs continuously because T cell production and exit in the thymus, from the thymus, steadily goes down. So this homeostatic proliferation occurs constantly, and this constant slow rate cell divisions eventually or actually are going to cellular senescence. It's called replicative senescences. So it's inescapable events. And the T cells eventually go into deeper state of cell senescence. And end stage of that is irreversible senescence of the T cells, which we consider as represented by the SAT cells. Those are very much extensive homeostatic senescent state of CD4 T cells, in this case, particularly CD4 T cells, as a consequence of continuous homeostatic proliferations. Okay, and B cells also goes into similar state of cell senescence, although the exact mechanism still remains to be seen. It could be replicative or it could be other reasons. But again, at the very old stages, you have continuous expansion and accumulation of aged T cells and B cells, which express unique molecule other than T cell receptors. That is CD153 on T cells and CD30. CD30 is a receptor of CD153. CD30 on these aged T cells. And today's theme is that those T cell, B cell also interact. When aged T cell meets B cells, somehow T cells are very aberrantly activated. And although I'm not showing here, if you have genetic knockout, either of CD153 or CD30, you have very much compromised development of both SAT cells and ABCs, implying that in this aged stage, there are still TB interactions, which has significant effects on immune conditions. So throughout life, there are TB interactions, although in each stages of age, a distinct context of the nature of the interactions. So I'm going to talk a little bit about the third type of TB interaction, what happens. Here's several data. It's actual data. As I said, these SAT cells, senescent T cells here, rarely respond to optimal T cell receptor stimulations, which induces robust, otherwise robust stimulation of regular T cells, as shown here. But if you add anti-CD153, which mimics the engagement with CD30, now even those SAT cells, senescent T cells, do show cell divisions, although not fully comparable to regular T cells, but still they show significant proliferation or cell divisions. And very unexpectedly or surprisingly, we found that this CD153 molecule is capable of association with every chain of CD3 complex, which is normally associated with T cell receptor as a signaling complex. So this finding was very surprising, and the molecular interaction of CD153 and CD3 complex actually reproduced in a non-T cell host by co-transfection experiments, indicating that this association of CD153 and CD3 complex is independent of T cell receptor. So this was very surprising. Then we examined what this CD153 molecule does during the activation of these SAT cells. And here's typical pictures. If you stimulate normal T cells with T cell receptor antigen, there is rapid activation of T cell receptor signaling by CD3, which is followed by quite swift internalization of the T cell, engaged T cell receptors intracellularly, followed by degradation, because this signal, effect of this signal is very profound. So this internalization occurs to prevent sustained signaling, or rather limit the signaling durations. And this internalization of T cell receptor complex upon the ligation with T cell receptor occurs very much normally, even in SAT T cells. But in the presence of additional presence of anti-CD153, you strongly inhibit the internalization of the engaged T-cell receptors. And consistent with this result, if you look at the activation of proximal ZAP70, which is a crucial kinase to transmit the T-cell receptor signaling, as I said, SAT cells show little activation of this ZAP70 kinase by regular T-cell stimulation. But if you add anti-CD153, you see very strong activation of ZAP70, which surprisingly lasts long for as much as 90 minutes after stimulation. Although not shown here, in regular T cells, this ZAP activation occurs quickly, but response subsides normally within less than 30 minutes. So the effect of this CD153 ligation is to prolong the signal to overcome this unresponsive state of SAT cells due to the senescence. So our hypothesis, this is a working hypothesis, and if those SAT cells meet regular antigen-presenting cells, like normal B cells or other antigen-presenting cells with foreign antigens, they are engaged, but those T-cell signaling cannot be transmitted downstream because those are senescent T-cells, so there are intrinsic hindrances to inhibit cell division, such as this micro RNA reduction, which results in a very high intracellular level of phosphatases and high levels of setting of CDK inhibitors, and also so-called senescence-associated heterochromatin foci, lots of heterochromatin. So those are programmed to inhibit or restrict those kinase signaling. So there is no detectable signaling here by regular T-cell receptor stimulation. Although the degradation process per se occurs normally because this internalization of the engaged receptor is now considered to be initiated by the dissociation of CD3 chain called CT3 zeta. So this has very high turnover rate. So upon stimulation, this CD3 zeta dissociate, and this results in internalization and exposure of lysosomal targeting, so on and so forth, and they eventually degraded to inhibit or to limit the signal strength. So this is explains unresponsiveness of senescence-associated T cell to normal antigen. But when these senescent T cells meet or encounter senescent B cells like this, ABCs, they now express CD30. And also, they have very potent antigen-presenting activity of self-antigens, any self-antigen. So this is polyclonally occurring events. Anyway, so again here, the T-cell receptor SAT cells are engaged by self-antigens. And normally this signal is not transmitted. But if you encounter this type of B cells, here is engagement, co-engagement of CD153 with CD3. And this 153 is associated with CD3 complexes. And what is probably happening is that this T-cell receptor complex tend to be internalized by dissociation of CD3 zeta. But this 153 CD3 complex is very, very unique, and they come to the T cell receptor nearby, and by cis interaction leads to the sort of providing CD3 zeta to inhibit the dissociation and thereby stabilizing this T cell receptor on the surface by restricting the internalization, which causes, as I mentioned, a very sustained signaling through this T cell receptor complex, which now enable to overcome these hindrances of senescent cells to cause significant activation or significant signal transduction. But to be noted is that although the signal is transmitted, the programming of the cell per se, downstream of T cell receptor, is already changed due to the senescence processes. So the real effect or final effect of this significant T cell receptor signaling now goes to the senescence-associated secretory phenotype rather than regular immune responses producing, for instance, interleukin-4 to kind of regular immune interferons. So this, we call it antigen-induced senescence-associated secretory phenotype. That is, senescent functional features are triggered or induced to be amplified by the T cell receptor signaling. This is very unique situation, we believe. Almost time. So here I simply summarize the T cell receptor T cell B cell interactions in the immune system. As I mentioned, T cells and B cells always function in the immune system cooperatively whole life, but the context is different depending upon the major context shifts with age. And in younger phase, this causes optimal regular immune response, which is clonal with robust proliferation and very strong activation of immune responses, antibody production, inflammation, and so on and so forth. But those are antigen-specific, not random. But as individual gets aged, you have to keep the number of cells. So it's immune homeostatic, not immune response. So by taking advantage of self-reactive, intrinsic self-reactive to T cells, now they start to divide slowly just to compensate the reduced numbers of T cells and to maintain the cell numbers. And here, the major purpose is to proliferation without causing any potentially harmful immune responses. So this is second stage of TB interaction. And final stage is, as I mentioned, aged T cell meets. It's not occurred in regular situations. They are circulating. But this particular encounter can occur when tissues are stressed by many different types of stresses, metabolic stresses, genetic stresses, or other types of physical stresses, which I come back later a little bit. And here, the PD-1, PD-L1 system is important to restrain the immune response magnitude. And here, those combination of CD153 and CD3 involved to sort of reactivating senescent cells. Oops. And we previously reported this system can work in sometimes in cancer systems, which turned out to be very effective. One of the effective ways of controlling human cancers are checkpoint blockade analysis assays. So we reported this almost 20 years ago. And we're happy to see that these are contributing significantly to the control of very much progressed, advanced cancer patients. So now we are questioning whether this interaction may have some effects on disease phenotype, and if so, if we can block this interaction, we may be able to sort of control the several age-associated chronic inflammatory diseases. And I show you one typical example here. And this is lupus model. And this particular types of lupus model, female lupus mice, spontaneously develops first anti-double-stranded DNA antibody, followed by glomerulonephritis, proteinuria, and renal failure, and eventually multi-organ cell death. So this is very much spontaneous autoimmune disease model. So we decided to examine the blockade of 153 and 30 interactions using a particular antibody which we produced recently. So if you start the treatment before the disease onset, this is set one, it's prophylactic treatment. And as you can clearly see, by the administration of this antibody, you can almost completely inhibit development of anti-DNA antibody or anti-nuclear factors, so if you like. And eventually, you can also inhibit the development of typical immune complex mediated glomerulonephritis, as shown in that picture. And even after the treatment, after the disease onset, we can still have a significant effect by this therapeutic treatment model and set two. And if you continue to treat these mice with antibody, after the antibody production, you can significantly inhibit overt proteinuria and also very much improved survival rate. So at least in some disease models in mice, though, these intervention of the interaction of aged T and B cells can really improve very characteristic disease, lupus. This is very akin to really human SLE. So immune aging can have actually dual phases, Janus phase we call. And in the first phase, you have a gradual increase of senescent state, or I would rather say deepening into the senescent state where you have progressive immunodeficiency. So that is one phase, and this should be certainly affecting the reduced, increased susceptibility to infection and possibly to cancer. And if it goes further, you have senescent cells, and in certain tissues where those T and B cells, senescent T cell, B cell encounter induced, you have very strong tissue reactions, inflammatory disease or autoimmune diseases. And as I mentioned, those signals are very characteristic in that CD30 and CD153 interaction induce mutual activation. So you constantly have feedback. I mean positive amplifying loops. So once this interaction is initiated in certain tissues, it goes on and on and on. And that is represented by very much persistent chronic inflammation. So those are apparently contradictory types of phenotypes associated with senescence, I think. So this is final slide. When you say immune aging affects disease phenotypes, there are two distinct phenotypes. One is due to impaired acquired immunity, which may cause to the increased risk of cancer progression as well as infectious diseases. And indeed, a recent paper by Palmer from UK very strongly suggests that thymic involution and the increased or rising frequencies of various types of cancer and infectious diseases like pneumonia are very much correlated. So there must be some linkage. So this is one part. And the other part is that due to this accumulation of senescent cells. And in certain situations, for instance, what we already reported is that in a high-fat diet, you see the accumulation of SAT cells and ABCs in the adipose tissues, where they expand lots and causes chronic adiposity, which leads to the increased insulin resistance. And we also see similar accumulation of aged T and B cells in the kidney under the renal hypoxic condition, only in the kidneys. And this leads to the progression of chronic nephritis, eventually leading to chronic renal failures. And more excitingly, although I'm not supposed to say this, difficult, but in certain very popular CNS diseases, you see massive expansion or accumulation rather of T and B cells at the same time, where they induce very much chronic oscillating neuronal diseases. So we believe that those aged T cell B cell interactions should be involved in that. So my final message is that by immune intervention, by particular antibody or whatever, you may be able to control age-associated chronic common diseases, irrespective of the disease types. That may be a very common basic way of control those common diseases. Okay, that's all my talk. And my scientific talk was almost, the last time was 1 year ago, so I'm not really doing these types of presentations. I'm not sure I can do it better. But anyway, I did my best. And those are my colleagues. And also, this research has been done with collaborative research funds from several pharmaceutical companies, including Sumitomo Pharma. Thank you very much.
Yeah. So thank you, Dr. Minato. It was wonderful to see you here, not only by Zoom, after so many couple of years. And second, thanks for sharing this very exciting research. So we have time for 1 minute, for one question. And perfect, Masa.
Thank you for exciting talk. ZAP70 is activated under CD153. So even without blockade antibody, ZAP70 inhibitor can cure SLE or other age-related diseases, do you think?
But if you do that, you inhibit all the immune responses. That's impossible. You don't want to lose infection resistance, right?
Okay, thank you. Yeah. Exercise and good diet is also still important. Thank you.
Thank you very much.
So the last science presentation will be Mitch Cronenberg. Mitch, since I came here a few years ago from NIH, is our wonderful partner in terms of immunology. So he has very close to the Cancer Center, only one block. He has been the director of the La Jolla Institute of Immunology for many, many years. Recently, he now he's the chief scientific officer from LGI. He has made many seminal discoveries in the area of innate like T cells, the regulation of mucosal immunology, pathogenesis of inflammatory bowel diseases, and many different areas. He has received many awards that probably in recognitions they will take a lot of time to just describe them. And what he will be presenting is really at the intersection, in this case, of fundamental immunology, autoimmunity, and cancer. So thank you, Mitch.
Thank you very much, and thank you for the invitation to speak here. And I'm going to talk about innate like T cells, which may seem like a contradiction if you're familiar with immunology, but I'll explain that. One of the key features of the immune response is immune memory. And what we know is that if your immune system sees, if your immune system sees a pathogen, clonal cells that recognize that pathogen expand. Some of them last a long time. And if the pathogen's encountered again, the growth of the pathogen is diminished, as you can see in this slide. And this is a property of adaptive immunity. This is a property of the T and B cells that Minato-san just described for you. But there are many cell types in the immune system, as you all know. Even immunologists find immunology to be complicated. And of course, there are innate immune cells, and there are innate immune cells in the myeloid lineage, but there are also innate immune cells in the lymphoid lineage, including NK cells, which were mentioned briefly earlier, and helper versions of NK cells that are called ILCs. But what I'm going to talk about today are innate-like T cells. There are really three types of them: gamma delta cells, NKT cells, and mate cells. And I'm going to be talking today about the NKT cells and the mate cells. So these cells have really different properties. First of all, we tell our students that T cells recognize peptide fragments, but these cells actually recognize non-peptidic antigens that are found in microbes, but there might also be cell versions of these. And these antigens are presented, just like peptides are presented by the polymorphic MHC molecules, HLA molecules that Dr. Yamanaka mentioned in the first talk. But these antigen-presenting molecules are not polymorphic. So we all have the same CD1D, which works for NKT cells, and MR1, which works for the mate cells. So these invariant antigen-presenting molecules have corresponding T cell receptors that are relatively limited in variability, particularly the alpha chain, one of the chains of the T cell receptor, is not variable. Now, what's innate-like about these cells? First of all, they make very rapid effector responses. So normally T cells won't make cytokines like gamma interferon unless they've seen antigen in a lymph node and undergone rounds of proliferation and differentiation. These cells, by contrast, make very rapid effector responses. And also, they can be activated even without engaging their T cell receptor, just by cytokines similar to NK cells. And this just shows you the antigens. The NKT cells recognize glycolipids. One of them that we found from strep pneumonia is shown. And the mate cells recognize riboflavin metabolites that are really microbial signatures. And many years ago, Laurent Brosset in my lab, he's now at Brown University, he showed, and others have shown as well, that this antigen recognition system is highly conserved. So a mouse NKT cell or mate cell sees the same thing as a human NKT cell or mate cell. And in fact, there's interspecies cross-reactivity. This is astounding because we diverged from the rodents 60 million years ago, and yet this specificity is conserved. By contrast, my influenza-reactive T cells probably won't work in your body because we have different HLA molecules. Okay, so because of this conservation of this specificity, these cells will not cause graft versus host disease. One size would fit all if they could be adapted for immune therapy, and various companies are trying to do that, for example, for NKT cells with special glycolipids, or using the cells as a platform for chimeric antigen receptors and so on. And I should tell you that I do consult for companies that are trying to weaponize, if you will, or use NKT cells in cancer immune therapy. And the most encouraging thing we have so far, in my opinion, is using NKT cells as a recipient for chimeric antigen receptors, because they could be off the shelf in the sense they won't cause graft versus host disease, and they persist and like to go to tissues. In considering how CD4 T cells function, one of the key findings in recent years is that there are different cytokines that CD4 T cells can make, and that cytokine production is driven by key transcription factors. So for example, if we consider gamma interferon, there's a transcription factor called TBET that drives gamma interferon. And that type of immune response, very useful for viruses or intracellular pathogens, is called a Th1 response. And the Th2 response, by contrast, which is characterized by different cytokines, by IL-4 and IL-13, is very useful for expelling parasitic worms and also for causing a lot of allergies, as you may know. Well, we found that for the NKT cells, there are also subsets that are like this Th1, Th2, and there's a third principal subset that makes a cytokine called IL-17, which is driven by a different transcription factor, RR gamma T. So we were able to go to a mouse thymus, which has about 500,000 of these mature NKT cells, and we could sort them based on specificity, and then using other markers, we found that there were different populations. In other words, there's a population in purple here that has a lot of RR gamma T, but very little TBET. And those are IL-17 producing NKT cells. So there are NKT17, NKT1, which by contrast have a high level of TBET, and then the blue ones are the 2s. And when we did bulk RNA-seq, there are triplicates here. These cell types were greatly different in their transcriptome. So you have a cell type which they all essentially have the same specificity, but they grow up in the thymus not as naive T cells that are waiting to see antigen, but that already apparently have some kind of gene program that drives very, very different effector functions. And in fact, we looked at these cells not only in the thymus, but we looked at them in peripheral organs as well. And what we did is we separated them again based on their specificity. So they're all NKT cells, and then on other markers. And then, so we call them 1s, 17s, and 2s. This color scheme continues. And we sorted them from different organs. And what we found by ATAC-seq and by RNA-seq is that the key thing about these cells was whether they were making a Th1 response, a Th2 response, or a Th17 response. And this just, at the bottom, it just shows you some of the ATAC-seq data that these functional cell types are very different despite having the same specificity. And it's almost as if they were preset or fixed. In other words, the idea is they grow up in the thymus for reasons that we don't fully understand. Some decide to make the type 1 response against intracellular pathogens. Some make the type 2, some make the type 17, which is very good for extracellular bacteria like strep pneumonia and others. So the question is, are these cells fixed or not? And it turns out they're not. So just, they seem to have a memory-like component, and that's really the theme of what I want to tell you. And even, I have to say, even for innate immune cells, even for myeloid cells, people talk about trained immunity, which means long-term adaptations that can occur after activation. So what we see is that we give mice a glycolipid, which is a potent antigen for these NKT cells. And what we can see is that they change. So here, this is a flow cytometry experiment. These are all NKT cells that you can see in the histogram. And they don't express these two proteins, PD-1, which you've heard about, and CXCR5. But after the antigenic stimulation, you can see that a lot of these cells have PD-1 and CXCR5. And if we look at these cells in particular, they have expression of a transcription factor called BCL6. So these are not factoids, but rather these are markers for another T cell subset that's called a follicular helper. These are T cells that go to the germinal center and are particularly effective at helping B cells secrete antibodies. So not only are there NKT1, 2, and 17, but there are also NKT follicular helper. And we and others have shown that these cells, in fact, are very effective at helping B cells secrete antibodies. But then there's this other population. So are these cells that maybe nothing's happened to them, or maybe they haven't seen antigen, and that isn't the case. It turns out they're also different. And so we have ATAC-seq and RNA-seq experiments. And what I can show you is that these guys that didn't become the follicular helpers, they have a very different chromatin from the follicular helpers. So the black and orange symbolize open chromatin sites that are different between these populations. And the RNA-seq also indicates that these cells cell types are highly different. And in fact, they're different from the so-called naive NKT cells, even though they're not really naive because they already look like antigen-experienced cells. And this just shows you that both populations, both the follicular helpers, which are in orange, and the ones that didn't become follicular helpers in black, which actually are very effective killer cells, as it turns out. Both of these populations are highly different from the cells, the NKT cells that were present before. And I won't go through the data today, but they last a long time. So the lipid antigen has a half-life of around 18 hours in the body, but these cell types persist literally for weeks. So the idea is that NKT cells can acquire separate effector gene expression programs in the thymus, which most T cells will only do after they've seen their cognate antigen in a lymph node. And there are similar gene programs in peripheral sites, but in a way, these cells, even though they're effector cells, are capable of further differentiation to a memory-like phenotype. And we've done some cell lineage tracing to ask, which one of these subsets becomes a follicular helper and which one becomes an effector. And I won't go through all of that data now. Okay, so what about the mate cells? The story for the mate cells, and mate means mucosal-associated invariant T cells. These are the cells that see riboflavin metabolites. These are about 2% of your lymphocytes in the peripheral blood. That's the average in the cohort that we've tested. And they're about 30% of the intrahepatic lymphocytes. So these are a very prominent population in human beings. And these cells also have in the thymus an effector function, so not quite as good as the spleen. But if we just concentrate on the mate cells in the thymus, some of them have the ability to make gamma interferon, and some of them have the ability to make IL-17. So in the thymus, there are mate 1 and mate 17 cells. And we can ask, is there memory? And in this case, we use bacterial infections. And what we found is that there are different bacteria. We started using Francisella tularemia, but originally, based on work from Australia, an Australian group, we used Salmonella enteric vaccine strain called BRD 509. And when we actually do an inhalation infection, so a lot of the work I'll show you is from the lung, and the mate cells in the lung in response to this bacteria expand dramatically from a few thousand cells to 50,000 or more, and they're present for 9 weeks. The bacteria are cleared in about 2 weeks. And so here again, we have something that looks like a memory response, that the cells, after seeing antigen, are long-lived. And in fact, if we reinfect with Salmonella, these mice are protected, so they have fewer CFUs. And if we infect with a different bacteria, in this case strep pneumonia, we can see that, and this is actually this is actually in a transfer experiment. So in this experiment, we expanded the cells with one bacteria, with Salmonella, and we took the so-called mate memory cells, transferred them to a different mouse, and then even 2 days later, you can see that the CFU are decreased by many logs. This works across bacteria, because different bacteria make the same riboflavin metabolite antigen for these cells. So we've tried to analyze what's going on with the mate cells, and we've done the single-cell RNA-seq experiment. And what you can see here is that these are the cells that never saw antigen. We don't get as many of them. These are cells that saw antigen. They fall into about 8 clusters. Some of them have more of a circulatory signature, some of them more tissue. So these in brown here, and some of them have more tissue residency. So there's heterogeneity in these cells as well after immunization. And we do have gene signatures which look like Th17 cells. In other words, the gene sets that would indicative of IL-17 production and gene sets that are indicative of gamma interferon production. And we have markers for these populations. So this protein marks the mate cells that'll make gamma interferon, and this one more or less marks the mate cells that are better at making IL-17. And the flow cytometry shows you that, that these two populations are mutually exclusive. The ones that have KLRG1 don't have IL-7 receptor anymore. And this just shows you, I can skip this, that there are mate-1 cells in the beginning before immunization, but the mate-1 cells after have increased expressions of certain genes like NK receptors, granzymes, and other things that might be indicative of their enhanced function. So I indicated those with that little circle there. So these two cell types have many differences, these memory-like mate cells, in terms of where they are in the tissue and other features, certainly the cytokines they make, but also their metabolomic or their requirements for metabolism. And just summarizing a lot of work, the MATE17 cells are very good at taking up lipids. So here's the MATE17 memory cells, here's the MATE1s. And they have increased mitochondrial activity, which we know from flux experiments, but here I'm just showing you mitotracker staining. So the cells that are MATE1s that have KLRG1, they tend to have lower mitotracker staining. In other words, less mitochondrial activity. And by contrast, gamma interferon production, which is a MATE1 function, is highly dependent on glycolysis and glucose uptake. Human mate cells from blood are also different metabolically, and the slide is a bit complex. I apologize for that. But on the right here, these are all human mate cells, and these are human CD8 T cells. And you can see the human mate cells, these 3 clusters here, are better at fatty acid uptake and fatty acid storage, comparable to what are called tissue-resident memory cells, but different from all the other CD8 T cells that we could see. So then the last thing I want to tell you is that's a fact. Is the fact interesting? In other words, does metabolism influence the generation of these subsets? And so we looked at VHL deficient mice. So when you delete VHL, HIF1 goes up, and you get more of a glycolytic phenotype, and that's shown here. For example, the mate cells are capable of taking up more of a glucose analog. And the control for that is the double knockout, if you will, of both VHL and the HIF1 target. So when we do the double knockout, the effect goes away. So that just shows you that there's a metabolic effect that's on target. And what this graph is one of several, and it's one of several mutant strains we looked at. What this shows you is that when you increase glycolysis through this VHL mutation, you change the subset distribution. And so here you can see that this is the litter-mate controls without the knockout, and this is a T-cell specific knockout. And you can see that the KLRG1 cells go up when you increase the glycolytic pathway, but this is dependent on HIF function, and the other population goes down. So just to summarize in my last slide, these innate-like cells do have memory, or some people don't like that, so we can call it antigen adaptation, meaning they change long-term after being exposed to antigen. But we also see similar things in the myeloid population. And in going into more detail, there are new functional subsets for the NKT cells that I showed you. For the mate cells, it seems that these cells don't change their function, but their function becomes enhanced. And these are long-term effects. And finally, that these cells have different metabolic choices, if you will, and metabolism affects their number, the mate-1 cells being more dependent on glycolysis, and the mate-17 on oxidative phosphorylation. There you go. I'm 5 seconds within the red light, and I'll thank you for any questions you might have.
So, thank you, Mitch. Any questions from the audience? So, do these cells play a role in cancer?
Yeah, so so there is evidence in colon cancer, neuroblastoma, and a couple of other cancers that NKT cells increase and are potentially protective. There are similar data for the mate cells, but there seems to be a less certainty about whether the mate cells are actually beneficial or not, and that really needs to be worked out. And the clinical, I have to tell you that people have been studying the NKT cells for a longer period of time, and clinical trials, not only in the United States, but there are a number in Japan. And mate cells have really, are more recently analyzed. Once people found the antigens and could make what are called tetrameric reagents to detect them, the study of these cells greatly increased. And so clinically, the study of mate cells is lagging behind quite a bit NKT cells. But there are people trying to develop mate cell-based therapies. The other difficulty with mate cells is that the metabolites of riboflavin that activate them are unstable. And that's made it difficult for people to, people are looking for more stable ones that could be used to activate the cells.
Yeah, so you will expect that it will be linking to some extent the microbiome to disease outcome, like including cancer and many others.
Yeah, certainly. And in fact, both of these populations are affected by the microbiome, and mate cells even more so. In the germ-free mouse, there are no mate cells. So they're highly dependent on the microbiome and highly dependent on different aspects of the microbiome in different colonies. And so that could be a link between the well-known effects of the microbiome on various kinds of cancer immunotherapies.
John. Yeah.
So, Rich, thank you. And I was going to ask a similar question to what you just answered.
John, the microphone's coming if you, sorry. Yeah.
I was, the last answer you gave about the MAIT cells was part of my question, but I thought you said about 2% of all cells were mate, and there was 30% of the liver. Did I hear you correct?
Yeah, it's astounding.
What's the liver issue?
We don't know. What I can tell you is that NKT cells in mice are also about 10 to 30% of the intrahepatic lymphocytes. And NKT cells and mate cells in mice and humans, even though I stressed how similar the specificities are, they're kind of inverse. So mice have a lot of NKT, and we have a lot of mate cells. And for some reason, one population or the other is important in the liver, whether this relates to bile acid metabolites, other aspects of the microbiome. The liver, of course, is, the circulation is from the gut is draining the liver. Exactly how that all fits together is not known. We know certain molecules that are involved, like CXCR6, for example, is probably important for this localization in the liver, and CXCL13 is ligand, but what this means biologically is not so clear. But yeah, it is astounding, and one thinks, you know, in mouse models of NASH and other diseases, NKT cells play a critical role. So one has to think that in the human counterpart, that mate cells have to be important. It would be astounding if they weren't. Yeah.
So, perfect. So thank you so much, Mitch and Dr. Minato, for these wonderful presentations, and returning to Kaoru Kitajima. So please, thank you.
Thank you, Silvio. And I'd like to, I have to say, we are listening to amazing frontiers, and we are coming to the closing keynote, which will be chaired by Dr. Masatoshi Hagiwara, and he's the Director General of Innovative Support Alliance Life Science and Director of Kyoto University Research Center in San Diego, right here. Dr. Hagiwara, without further ado.
Okay, thank you, Carl. So I would like to introduce the last speaker of this symposium. Professor Michinori Saito is one of the most excellent professors of our medical school. He has investigated the development process of germ cells, and recently he succeeded to show that we can produce matured sperm or oocyte from iPS cell, and they are fertile. I think his science papers gave big hopes to infertile couples. So, and his laboratory is located in the same building with us, and my laboratory is on the third floor, and his laboratory is second floor. In this meaning, I'm always upper pass of him. Thank you so much. So please, Professor Saito.
Hagiwara Sensei, thank you very much for a kind introduction. And it's really a great pleasure for me to join this meeting. So I think since the audience are quite diverse, I think today, I'd like to briefly provide a kind of brief overview of our research on germ cells, and also introduce recent activity by the Kyoto University to promote the study of human biology. So germ cells, the spermatozoa or oocytes, and their precursors are a source of totipotency, thereby ensuring individual development and also heredity as well as evolution. So we have been very much interested in the genetic as well as epigenetic mechanisms underlying such unique and also essential functions of germ cells, such as epigenetic reprogramming, meiotic recombination, and also very much high fidelity of genome replication. So this is a brief summary of mouse germ cell development. So the germ cell lineage arises as primordial germ cells, or PGCs, early in development. Interestingly, PGCs are specified much earlier than the like gonads or testis or ovary primordial specified. So they initiate migration toward future genital ridges. And during such processes, they undergo a so-called epigenetic reprogramming, which include genome-wide DNA dematuration, leading to imprint erasure in both sexes and also X chromosome reactivation in females. And this process also involves dynamic changes of genome-wide histone modifications. So once primordial germ cells reach embryonic gonad, They are called oogonia in females, or maybe a spermatogonia in males. They have acquired a kind of epigenetically identical paternal and maternal alleles. That's the only time point in our life we acquire such sort of epigenetic equality of the parental chromosomes. And when germ cells colonize the male genital ridges or testicular primordia, they receive signals from testicular somatic cells to differentiate into spermatogonia by acquiring androgenetic epigenome. And in contrast, in the embryonic ovaries, they receive signals to differentiate into oocyte and immediately initiate meiotic recombination. So the germ cell lineage repeat this kind of epigenetic reprogramming and programming cycles to ensure a kind of totipotent epigenome. So based on the understanding of the mechanism of mouse germ cell specification, we have shown, it's already more than 10 years before, starting from mouse embryonic stem cells or iPS cells. They can be induced by a stimulation of these cytokines into the epiblast-like cells, which are the precursors for all somatic cells as well as germ cells in vivo. And when dissociated into single cells and stimulated by cytokines including BMP4 under kind of floating aggregate conditions, many of these cells start to express key germ cell markers such as BRIMP1 or STELA. And these cells, which shows gene expression and also the epigenetic property, very similar to mouse primordial germ cells. So we named these cells as PGC-like cells. The remarkable point of these PGC-like cells was that when FACS sorted and transplanted into the testis of neonatal mice, they spontaneously contributed to spermatogenesis to form spermatozoa, and they contribute to their fertile offspring. When starting with XX female ES cells or iPS cells, we can also induce PGC-like cells in the same manner. But in this case, when dissociated or isolated PGC-like cells are aggregated with mouse embryonic ovarian somatic cells to form so-called reconstituted ovaries and then transplanted into ovarian brusa, these PGC-like cells differentiate into the oocyte-like cells, which are also, upon in vitro maturation and fertilization, contribute to the fertile offspring. So these findings indicate that under the in vitro conditions, we can induce functional precursors of both oocytes and spermatozoa. So this experiment shows that we can induce these PGC-like cells in an abundant number in the culture dishes. So based on this kind of system, for example, it has been possible to really demonstrate a very detailed mechanism of transcriptional or signaling control of germ cell specification, as well as to delineate chromatin or nucleome programming during germ cell development. And later, Hayashi-san's group in Kyoto University has succeeded in continue to culture reconstituted ovaries in vitro to form a functional oocyte in vitro, so that all oogenesis processes can now be recapitulated in vitro. And then these kind of oocytes contribute to the offspring. We have also shown that when PGC-like cells are aggregated with testicular somatic cells to form reconstituted testices, these cells contributed to spermatogonia and then sperm, so that also these spermatogenic pathways can be reconstituted all in vitro, at least in the mouse. We have also shown that, for example, PGC-like cells can be expanded under in vitro condition without the use of embryonic gonadal somatic cells, so that we can understand, for example, under kind of defined condition for the mechanism for example, female germ cell determination. So we are now in the process of translating these findings in the mouse to humans. So this is the scheme of human germ cell development. As you will see, the key event that happens during human germ cell development are highly conserved with that in the mouse. So PGCs are specified early in development. They undergo epigenetic reprogramming. And depending on the sexes, they are specified either into spermatogonia or the oocyte. But of course, there's key differences. For example, as a matter of course, human development takes much longer time than the mice. And maybe due to that, the developmental transitions are kind of very asynchronous. For example, in the newborn female babies, there are primordial follicles completed the first phase of the meiosis, but at the same time, oogonia, a very immature oogenic progenitor, kind of remaining. And also, humans use divergent signaling, transcriptional, and also metabolic regulations. So still, it may be possible to say that the mechanism of human germ cell development remains largely unknown. Also, about 10 years before, to tackle this kind of point, because human embryonic tissues are very difficult to use, by using cynomolgus monkeys as model organisms, we begin to work on the mechanism of primate germ cell development. So this is a very simple phylogenetic tree of primates. We are kind of humans. And these great apes are the primates most closely related to humans. But of course, we cannot use them as a model organisms. And these macaque monkeys, including cynomolgus monkeys and also rhesus monkeys, are primate species with which we can work in a kind of experimental manner. So using this animal, we have especially asked two key questions. So one is, what may be the developmental counterpart of a pluripotent embryonic or stem cells, and how germ cells are specified in the primate? So this is their photograph of very early post-implantation embryos of the cynomolgus monkeys. So this is the epiblast, very flat disc-typed, very similar to that of the humans, but very different from that of the mice, which are covered by the hypoblast as well as the amnion. And the trophoectoderm, which are just opposed to the epiblast in the case of the mice, but it is very apart from the epiblast in monkeys. So it's very similar to the human early embryos. So by analyzing single-cell transcriptome of these cells and comparing with that of pluripotent stem cells, we have shown that, for example, monkey development proceeds with this pre-implantation inner cell mass, progress through this pre-implantation epiblast. They, upon implantation, change into post-implantation early epiblast, and then gradually gastrulation starts, and then they also shift to post-implantation late epiblast cells. And interestingly, monkey embryonic stem cells, which are derived from ICM cells here, are acquiring a property highly similar to post-implantation late epiblast cells, which was kind of very different from the case in the mouse, where mouse ES cells are very similar to ICM or pre-implantation epiblast. And the second question was where PGCs are born. And surprisingly, what we have found by immunostaining was that these SOC17 and also TFAP2C double positive cells arise not from the epiblast like in the mouse, but from the amniotic ectoderm. So this was the kind of very surprising finding to us. So probably also in the humans, the germ cell lineage originates from the nascent amniotic ectoderm. So based on these knowledges, as well as mouse in vitro model systems, we have shown that the human iPS cells bearing two germline reporters can first be induced into incipient mesoderm-like cells. And then they can be induced under the floating aggregate conditions by stimulating with BMP4 into like a TFAP2C and BRIMPAN positive human primordial germ cell-like cells, bearing gene expression similar to human and monkey PGCs. And interestingly, when these PGC-like cells are aggregated with mouse embryonic ovarian somatic cells and culture under the so-called xenogenic reconstituted ovary conditions, these human PGC-like cells mature into this DDX4 positive so-called human oogonia or early oocyte-like cells, resembling in the morphology as very similar to reported human oogonia. And not just the morphology. If you look at the gene expression trajectories, human oogonia and early oocyte-like cells, although I don't show the data acquiring the gene expression property highly similar to the human immature oogenic lineage cells, and also not just with the transcriptome, but also epigenetic aspect. They undergo substantial DNA methylation reprogramming, acquiring very low level of genome-wide DNA methylation levels, very similar to those in the human germ cells that you observe in vivo materials. So human PGC-like cell-derived cells acquire transcriptome and also epigenetic properties similar to those of human oogonia and early oocyte. And this, indeed, xenogenic ovarian systems are kind of highly useful to extend to the other lineages, such as monkeys. And for example, you can also induce monkey, starting from monkey embryonic stem cells, PGC-like cells, and you can aggregate with mouse embryonic ovarian cell, somatic cells for further culture. And during such sort of culture, like 49-day plus 56-day culture, you would see that initially these PGC-like cells start to express TFAP2C, but repress it and gradually upregulate DDX4, and then DDX4 expression levels become very high. And if you fast forward these DDX4 extensively highly positive populations that has more kind of 4N chromosomes, and we found they are reaching at least to the zygote stage of the meiotic prophase. So now we can induce meiotic fetal oocyte in monkeys and also humans. So this is a kind of potential scheme for a human in vitro gametogenesis research from pluripotent stem cells. So this early phase has been reconstituted. And the obvious question is that whether we can induce these cells further into, for example, primary oocyte at the primordial follicle stage, and even then mature to make a kind of fertilization possible. So we have been working on this oogenic lineages currently. And to do this, although it is very easy to say just by words, but what you may have to do is that, for example, first, to develop a kind of culture conditions to mature immature oogonia into the oocyte. There was no such systems reported in humans and primates. And the success of mouse in vitro oogenesis really depends on the pre-existing culture to mature in vivo PGCs into the oocyte. So we thought it's a kind of very important challenge to explore the conditions to enable this kind of processes, first using the in vivo materials. And indeed, we have very recently reported that the human as well as monkey embryonic ovaries, when they dissociated into single cells and reaggregated as reconstituted ovaries, and after a floating culture under kind of certain conditions for like 3 or 4 months, these immature oogonia-like cells spontaneously differentiate, complete the meiotic prophase to differentiate into the diprotein oocyte with surrounding granulosa cells forming primordial follicle-like structures. So this is a case of human in vitro culture system, starting from mitotic oogonia, passing through meiotic prophase to reach diprotein phase. That is also the case in the monkeys. And this kind of culture is progressively updated, so that the now starting from this immature ovaries, we can induce many primordial follicle-like cells. So this kind of research has made a kind of prospect that in some days human in vitro gametogenesis may be possible. So currently, the germline cells, of course, only the in vivo cells we use for our reproduction. But due to the emergence of these iPS technologies, we can now induce primordial germ cell-like cells that originated from any somatic cell lineages that may be induced in near or maybe some future to spermatogonia or immature oocyte that can be used for fertilization, or maybe a kind of treatment of infertility, or maybe at least it's modeling. But somatic cells are highly different from the germ cells in their development, and having lots of genetic or epigenetic abnormalities arising during our maturity. So we are not really sure whether these in vitro generated human gametes are genetically or epigenetically normal. And also we have serious ethical issues if we really want to do that. So facing against this kind of situations, at least I thought it very important for us to really promote the very basic human biology, including facilitating our understanding on the human development, growth, and evolution, as well as heredity. So for that reason, the Kyoto University has established the second world so-called premier international research center initiative, second to this ISMS, Institute for the Advanced Study of Human Biology, which we call ASHBI. So a kind of theme and key research goal of our institute is what key biological trait may make us human, and how can knowing such sort of things lead to a better cure of kind of disease state. For that purpose, we are promoting the study of human biology with a particular focus on how the genome is regulated. And we want to clarify a kind of principles defining the species differences and also the human trait. And we are making primate models for various key intractable human diseases. And so we are aiming to reconstitute key human cell lineages or tissues in vitro. These activities is somewhat overlapped with what have been done in the ASILA, but we are working more, how to say, basic aspect as a kind of complementary research. And promoting human biology sometimes makes encountered kind of ethical problems, so that we want to contribute to formalizing an international ethics standard for human biology research. So this is current groups. So the institute constitutes of like 4 branches working on developmental biology and also genome informatics, making primate models and also basic and clinical medicine. And very importantly, we are performing a kind of fusion research with bioethicists as well as mathematicians to understand kind of how the genome information is really treated. And so we have 3 core facilities, such as single-cell genome information analysis core, and also primate genome engineering core, and also non-human primate phenotypic analysis core. And we have a hub building in the very center of the medical campus of Kyoto University. And many investigators, main laboratories residing nearby, so that we can really make a kind of tight collaboration to promote the further studies of human biologists. And I think it would be wonderful if we can make this opportunity to make some sort of collaboration with UCSD members. For example, Ueno Sensei is already having on-site laboratories here for working on human immunology. So this is our lab members. I thank all the members of our laboratory for this work. Thank you very much.
Thank you very much for excellent talk. So I can take several questions from audience. Yeah, please. Yeah, I give you the microphone. Yeah.
Yes. So this is obviously a very a real challenge for ethicists, and you know, there are people in the U.S. doing this sort of work as well, and they are searching for some kind of a solution, because this is it's probably the most important thing that people worry about that can happen with iPS cells. So how do you envision making this kind of idea acceptable to various cultures in Japan and the U.S.?
You mean the ethically for the
ethical issues, because
yeah, sure.
you know, it just anytime I bring this up to somebody, they are always kind of alarmed. So
yeah, exactly. For example, I have many emails, for example, from infertile peoples, whether it is now possible to make sperm oocyte, but it is very difficult to answer. At least still, it's a kind of very immature stage to do that. And even in the mice, the rate of success is extremely low compared to in vivo, for example, oocyte, like 1 in 1,000 or maybe 1 in 10,000. That means there are lots of epigenetic abnormalities in the in vitro produced oocyte, or in the case of sperm, it's more like, because we can make a lot, so it's a bit different. So at least I think it is still very, very premature, and much more basic research is necessary. But when in some future every technological hurdles are gone, whether we can use such sort of cells for our reproduction is a key theme. And for that purpose, I started to studying the philosophy of Japanese philosopher to consider whether there might be any potential differences, whether we are derived from somatic cells or germ cells. But according to some sort of philosophy based on Buddhism, the difference between somatic cells and germ cells are almost kind of nothing compared to the concept of universe or anything. So in some future, it might really become possible, especially for kind of infertile peoples.
it's going to be more difficult in the United States, I think, in the direction they're going right now.
Yeah, there's actually a discussion session happening in April in the US to discuss the current state of in vitro gametogenesis. And the panelists include scientists and also ethicists, and also the one working on law as well. So this kind of activity is kind of increasing now. Maybe next week we have it also in London. So.
Okay, please, yeah, please give him the microphone.
Thank you for the very lucid lecture, Professor Saito. I'm wondering, with the advances you've made, in order to apply this more broadly, you will be working with different individuals. And so the role of individual variation in the success of the various steps, do you have comments on that? Is is one mouse the same as all mice? Is one human the same as all humans? What's the challenges involved in applying this in an equitable way so that so that many individuals could be successfully used?
Thank you very much. So, for example, we are using several IPS lines derived from different individuals. And the induction efficiency, for example, into the primordial germ cell-like cells and further are really different depending on the cell lines. But we are not sure whether it is because of these genetic differences or the epigenetic differences that arise during IPS cell generation. And it really requires a lot of work to answer. But generally, I think, at least in the humans, it's not so difficult to induce germ cell-like cells, at least for the first 2 or 3 steps. Maybe if you want to do further, then there might be some further potential differences. But yeah, that's a current state.
Okay. No more questions? Oh, Professor Yamanaka.
So you have generated mice already from this technology. So once pups are born, do you see any abnormalities in those mice? At least for the mice born with like a transplantation, the first ones through a transplantation into testes, we don't really see any obvious phenotypes. And that was also the case for offspring born from the oocyte after transplantation. But if you look at like Hayashi-san's paper 6 or 7 years before, oocytes or embryos or the fetuses derived from totally in vitro derived oocytes having lots of strange abnormalities, probably stemming from epigenetic abnormalities during the embryonic stages, similar to like imprinting diseases or maybe compound loss of several epigenetic marks. So very strange phenotypes. So in the case of in vitro derived oocyte. So I think for oocyte, in case of oocyte, which really transmit many epigenetic information to the next generations, and also responsible for reprogramming sperm epigenome, it should be more kind of difficult to really achieve, to create in vivo-like things. Although I don't think it is impossible. So when making like new life from iPS cells, we need to worry about both genetic abnormalities and epigenetic abnormalities. So genetic abnormalities, they came from somatic mutation. But like 10 years ago, we generated many mice from iPS cells through chimera and germline transmission. And we observed those mice until they die. And actually, they were normal. Yeah, somehow. Yeah, in terms of longevity. If you use like c-MYC retrovirus, they often develop tumors. So that's not good. But if you do not use c-MYC retrovirus, or you just use like plasmid, those mice were just fine. Yeah. So I believe we don't have to worry too much about somatic mutation or genetic abnormalities. But you have to overcome epigenetic abnormalities. Definitely, that's what I'm also thinking. So, for example, recent paper on I think, proposing that their aging is epigenetic aberrations or something, claiming that we have lots of somatic mutations, but usually that doesn't, although it happens in a very critical genes, doesn't cause diseases, but this epigenetic balance seems to be more important.
may really suggest that that's more difficult things to control.
So since they are epigenetical modification, I believe you can do it. If it's genetic, it will be next to impossible. But as long as it's epigenetic.
Yeah. But at least we accumulate these mutations. So maybe we better to use very young cells for generating iPS cells, maybe as a precursor for in vitro gametogenesis, like a cord blood cells.
Yeah, cord blood cells is probably the best. If you have your own cord blood cells.
Okay, thank you so much, Professor Saito. I would like to close this session.
Thank you very much, Dr. Saito and Dr. Hagiwara. Now, I would like to move to a final remark, closing remarks to end this wonderful and exciting, session. And so I'd like to introduce Dr. John Carruthers, Vice Chancellor for Health Sciences here at UC San Diego. Please.
Oh, please don't clap for me. Clap for all the speakers. I'll just say, wow. This has been a very impressive symposium. First of all, I'll just say, for those from Kyoto, happy March, because I know you're jet lagged and it's actually March 1st instead of February 28th. So thank you for coming and bearing, and I know I'm standing between you, your reception, and ultimately some sleep. But I just want to say I'm very pleased with this wonderful partnership between us here at UC San Diego and Kyoto University. The collaboration seems strong, and I know there's opportunity for it being stronger. And I want to thank my predecessor, Dave Brenner, for helping initiate this. This was a wonderful, wonderful first joint symposium, incredible, with wonderful speakers, and representation from investigators from other parts of our UC system as well. I think this is a very hard thing to do, but unprecedented and going to be highly collaborative. It's also fantastic that it's on the site of UC San Diego, represents a lot here in our Sanford Stem Cell Symposium. Thank you for partnering with us. In your first two symposia, the joint symposiums in 2015 and 2016, there was a focus on regenerative medicine and cancer research. And my understanding is that from those initial symposia, it's led to multiple publications, particularly in the field of medical imaging, advanced research collaboration in stem cells, and we heard just a wonderful, several wonderful talks for spinal cord injury and treatment, as well as visiting scholars to the lab. So this has been fantastic. The other thing that has been wonderful has been the Research Institute, Kyoto University Research Institute in San Diego, in our Center for Novel Therapeutics that commenced in 2019, which allows deepening of our own partnership. In fact, I know our investigators from the Morris Cancer Center and the Kyoto University School of Medicine were able to maintain the momentum even during this thing called COVID for the last 3 years through many joint webinars. And my understanding is it attracted over 1,000 viewers each time. So that tells you how interested this partnership is. In my role as Vice Chancellor for Health Sciences, which I've been a total of 6 weeks, moving from the University of Michigan, I will focus on our overall arching strategy for all health sciences. We will be expanding our clinical services over time, our clinical enterprise. We will foster continued and growing research collaborations such as this one, and enhancing many of our educational opportunities. And we had a wonderful discussion with the Chancellor earlier today. For me, this is critical importance to marry many of our basic and translational sciences with our partners in Kyoto. And I just want to say on behalf of our own health sciences, as well as Chancellor Kosla, who's representing the entire UC San Diego, I want to thank President Naguro Minato for your partnership with our university and even our other California universities. Thank you, and I hope you have a wonderful reception.
Thank you, Dr. Carruthers. And as you see, I would like to thank the UC San Diego for its wonderful hospitality. And we are very happy to hear such an encouraging word from Vice Chancellor for Health. But we like to foster our partnership in health science, which is spearheading, and beyond. So with that, I'd like everyone to applaud another round of appreciation. Thank you very much.